Uh, yeah. Good morning, everyone. Good morning, all listeners and panelists. So I hope
Good morning, everybody. Now, I would welcome you all to the second day of this international webinar. And I would like to introduce to today's guests.
Anshuman, is there any problem?
Yes, sir. Uh, I would like to welcome our special guest and chair for today's session, Professor H.S. Gupta, sir, former Director General, Breeding Institute for South Asia, BISA, New Delhi. Dr. Gupta, an environment scientist in the area of crop breeding and biotechnology. He has been decorated with fellowship awards of several academic including the Rockefeller Foundation's Career Fellow and as former director, Indian Agriculture Research Institute, New Delhi, and former Director General, Borlaug Institute for South Asia, BISA, New Delhi. He is recipient of several prestigious awards with I.C.B.R.D Team Award, Dr. Rajendra Prasad Award for Best Book in Crop Sciences, NRDC's Meritorious Invention Award, Hari Om Ashram Trust Award, etc. We welcome you, sir, to this international webinar, Day 2. A huge round of applause on this virtual media for Dr. H.S. Gupta, sir. I would like to
Thank
introduce
you.
to you to the organizing secretary of this international webinar, Dr. P.K. Upadhyay, sir. I would also like to welcome Professor Rajendra Kumar, sir, the organizing chairperson of this international webinar. I would like to welcome the lead speaker for today's session, Dr. Lakhbir Singh. Dr. Lakhbir Singh, sir, is the professor at SRM University, Amravati, and postdoctoral Oregon State University, Corvallis, USA. Topic for today's session for Dr. Lakhbir Singh would be Microbial Electrochemical Technology Overcoming Water, Energy, Food, Environment Challenges. We welcome you all. We'll start now. I hand over the mic to Professor Dr. Lakhbir Singh, sir.
yeah. Thank you, Mr. Anshuman Upadhyay, for the introduction. So thank you all. Yeah. So I would like to thank organizers and Indian Society of Genetic Biotechnology Research and Development Agra for giving me an opportunity to discuss about microbial electrochemical technologies. But before start, I would like to thank panelists and listeners. Okay, so without taking any further time, I'm going to start my talk today. I'm going to share. Okay, can you see my slides?
Yes, sir. We can see your slide.
Okay, sorry.
Yes.
Yeah. Okay, sounds good. Okay, so yeah, so next 15 to 20 minutes I'm going to speak about microbial electrochemical technologies. Yeah, microbial technology is recently a new technology has been discovered in Penn State in 2005. So I'm going to discuss how we can overcome water, energy, food, environmental challenges using microbial electrotechnology. So as we all know that the water, energy, and food are essential for human well-being, for poverty reduction, and sustainable development. So this is the most essential elements for our environment, for our human sustainability to sustain life. So global projection, as we know that because of increasing population, the global projection include the demand, for example, fresh water, energy, food will increase significantly over the next decade. It increase significantly because of the pressure of the population growth and mobility, because of economic development, because of industrialization, because of trade, because of urbanization, because of technology change and climate change. So agriculture basically accounts for 70% of the total global fresh water goes to the agriculture sector. Uh, Agriculture's production and forestry, fishery, along with the entire agri-food supply chain, is used also to transport energy in different form. At the same time, the food production and supply chain consumed about 30% of the total energy consumption. So the energy, as we know, that is required to produce the transport. We need energy to transport the food, as well as to extract, pumping, transport, and to treat waters in cities, in metropolitan cities. So this situation is expected to be more worst in the near future because, hello, yeah, some sounds coming. So this situation is going to be worst in the near future, as we need 60% more food will be needed to produce to feed the world, because population growth is increasing day by day. So the total 60% more food we need up to 2050. So global energy consumption is also projected to increase up to 50% because of industrialization, because of transport sector. We are using huge amount of energy. Similarly, the water withdrawal for irrigation and projected to increase around 10% by 2050. So you can see the demand of food, demand of energy, demand of water is keep increasing. And also as the demand grows, there is increasing competition for the resource between water, energy and agriculture, so and other sector also, so which has unpredictable impact for our livelihoods and the environment. So here you can see that in the first slide, interconnection. So the food, energy, water, they are interrelated, they are interconnection. For example, for food production we need energy, we need water. For example, for energy production we need water, and sometimes like for bio fuels we use a large amount of grains, corn. So they are interrelated, and as the demand is increasing, it has the impact on our environment. So we need technology, where we need some alternative technology to fulfill that demands. For example, for water treatment, for example, for some other alternative source of energy production, for food production. I'm not going to talk about food production directly using this technology, but it could be we can generate some like fishery products, some aquaculture products using microbial electrochemical technology. So they are all interconnection, and they have a huge impact on our environment. So I'm giving you few numbers, how much energy we need to manage our water infrastructure. Water infrastructure means like for water cleaning, for water for agriculture sector, water for the water desalination, for drinking purpose. So these numbers for water related and data, I don't have Indian data, but I have some, because this study has been done in US, so we have some US numbers. But for later on, I will explain about lignocellulosic biomass. That data we have. So annual energy use for the water infrastructure is around 30 gigawatts. These numbers are not small, a huge number, which means that around 5 to 6% of all generated electricity goes to manage water infrastructure. Water infrastructure, maybe we need, because we need energy for desalination, for pumping, for agriculture. So similarly, energy used for wastewater treatment, I think the number is quite same in India also, because India producing huge amount of wastewater, and also in India approximately 80% of fresh water goes to agriculture sector. So energy used for wastewater treatment around 15 gigawatts. to manage wastewater, because we have to treat like sewage treatment, like septic tanks, and our industry wastewater, our hospital wastewater. We have to treat. We cannot simply dispose to the water bodies. So as you can see, the new source, the new energy source, wastewater. Wastewater could be the new energy source. we can extract we can extract energy from wastewater, especially mostly from the, because the in wastewater contain some organic matters or organic pollutants, so we can extract that energy. So instead of using direct energy to treat the wastewater, we can extract energy from wastewater and we can utilize for to manage our water infrastructure. So so as you can see here, so domestic and industrial wastewater contain around 17 gigawatts, which means that if we convert the wastewater, stored wastewater energy, we could get approximately 17 gigawatts. So domestic water contain most of organic matter, such as like some organic pollutants, because it comes from municipality, comes from houses and kitchens. So we can easily extract these energy and we can use to treat water. That energy we can use, energy produced from such type of sources we can use for our for daily use. So here, so wastewater contain totally 17 gigawatts. So you can see here number, so approximately 45 gigawatts energy we can save, because the wastewater contain amount energy 17 gigawatts, and also we need some additional amount of energy to manage the water. So cellulose biomass energy in India is approximately 50 million tons of agriculture waste is coming every year. So if we able to convert that waste into energy, we could get approximately, these number are not exact but approximately I'm saying, because it's very difficult to collect data uh, exact biomass, but approximately around 50,000 megawatts the energy we can get. So you can see how much energy we can extract, as well as with energy we can save energy and we can also treat water. So so now how we can extract these energy? There are many different conventional technologies are available, but some of them are environmental friendly, some of them are not environmental friendly, and some of them are cost effective, but some of them are not cost effective. So we need technology where the environmental friendly, cost effective, as well as simple, to extract huge amount of, I would say, waste energy, as well as the to treat wastewater and extract some valuable products. So so here the as you can see here, the electrochemical, microbial electrochemical technology. So microbial electrochemical technology, as I mentioned, that is the system, is the technology where microbes at one of the electrode degrade organic matters. Any type of biodegradable compound can be degraded by using the microbes. So these microbes exist on electrodes. So they can degrade uh, any type of biodegradable compounds such as wastewater or lignocellulose waste, and they can generate electrons. So as you can see here, the electron, they can generate electron, they can extract electrons and proton, and there's electron proton combine at the on the cathode and generate electricity. That's how this system works. This system is very simple. It has microbes which exist on electrodes, anode and cathode, and that's all. And we can use any type of biodegradable substance in this type of system. Of course, we need some type of chambers and to operate. So if you see, the definition is the presence of metabolic activity of electroactive bacteria. So we need some type of exoelectrons and microbial the bacteria that can use, they exist on solid electrode and they can oxidize different types of compounds, organic compound, which exist in the wastewater, to synthesize chemical bioremediation of pollutant matrix, as well as to treat contaminants. So here you can see the number of multiple products we can extract from this technology. Like we can get reclaimed clean water, we can produce energy. Energy could be in the form of hydrogen, in the form of methane, in the form of acids, in the form of ethanol, methanol, and also we can extract nutrients from the wastewater. Like as you can see, our agriculture runoff wastewater contain huge amount of we use nowadays fertilizer, so we can easily extract nitrogen, phosphorus from the agriculture runoff water, and then we can protect also eutrophications and some other value added products. Value added products could be like H2O2, ammonia, urea, and single cell protein, which can be used for our aquaculture as a animal feed or animal food. So a number of multiple products can be produced. So this system basically works same like we eat food, we oxidize it, and the we produce electron we send to the respiratory enzymes, and then once we're done, it goes to the oxygen, and that's how we eat and breathe. So similarly, in this system, we feed with the waste. The bacteria exist on electrodes, degrade that waste, generate electron and proton, and this goes to the oxidation and oxidize in another chamber, cathodic chamber, where they receive the electron, and as you can see that, and produce reduced oxidant and produce water. And so because of that, the generation of electron and proton, there is potential difference, and they generate. That's how when the voltage into current, it generates some current here, and that is the power. That's how we can generate electricity from this system. It's very simple system. So here, so as let's say you don't want to produce electricity, you want something else. For example, hydrogen. You want hydrogen. Hydrogen is a clean fuel, as we know that is highly has having the high calorific value, and it's clean fuel. When we burn hydrogen, it produces only water, which we can recycle again and again. So let's say you want hydrogen production. So what we need to do, we just need to remove the oxygen from the cathode chamber, and then no oxygen chamber in cathode. So here, but what will happen? There is no major change with the conventional microbial electrochemical system. This is the what we have to do. We have to remove the oxygen from the chamber, but what will happen? Actually, this reaction will not drive the electron, because bacteria is not capable to remove the extract the electron from the organic matter. So what we have to do? We have to use little bit additional source of electricity to boost the bacteria to extract the electron, and that's how our fuel cell also works. So electron comes here and proton, they combine in the cathode. That's how they produce hydrogen. So my work, most of the work I have done on hydrogen production. Later on I will explain this type of system we called microbial electrolysis cell. Basically we use for hydrogen production. We were able to generate around 8 to 10 liter per hour for 20-liter reactor hydrogen from like brewery wastewater and some other industrial effluents. And there is some other types of systems also we have. Let's say microbial fuel cell, microbial electrochemical cells. So this is the here I this one is the microbial fuel cell. The difference between microbial fuel cell and microbial electrolysis cell, only the cathodic chamber. In microbial electrolysis cell, we do not have oxygen. So that's why we need additional source of supply. This additional electricity is less than 1 volt. We could get from external source like from solar or from the renewable source of supply. Very less. It's very less to drive the reaction, to make it spontaneously. So this system is very simple. You can see here we have anodes. This is the anodic chamber. So we have cathode, and we may not have membrane, or we may have membrane. This system could be one chamber, two chambers. So but it got to be microbes everywhere. It could be microbe on anode. It could be microbe on cathode. So it depends what product you want to produce. So here, as you can see that this system can utilize any type of biodegradable substance to generate number of multiple products. So as you can see here, the acetate can be utilized, glycerol, acetate, glucose, and generate number of products from this system. So, in my research, I will explain a little bit. We worked on microbial electrochemical cells. Why we have selected this system? Because it has some advantages over MFC, because our target was to generate hydrogen, as hydrogen is clean fuel, as I mentioned earlier, and our target was to generate less than $2 per gallon cost. So this system has some benefits over other biological techniques to treat wastewater as well as to generate energy and some other valuable products. So, as you can see that the other technology for water hydrolysis, dark fermentation, aerobic, anaerobic digestion for hydrogen production as well as for wastewater treatment. So benefits over them is a low energy biological catalyzed technique, and also it has a high energy yield from organic matter and low operating cost, and also suitable for low strength wastewater. But still, this technology, the microbial electrochemical cells, still in his infant period, it still is an unmature state because of low hydrogen production rate, because of high capital cost. And the another disadvantages of uh, this this system is basically suffering from high internal resistance. So high internal resistance, because the resistance, if you see here, is associated with anode material, it is associated with cathode material, it also associated with membrane if it is there, and also associated with solution. Solution means it could be effluents or other part. So the internal resistance is the big problem, which ultimately hindered the application of microbial electrochemical technology at large scale. So what we have done, we have designed reactors, hybrid electrochemical reactors to solve this problem. So how we solve, I will explain one by one later on. And another problem in MEC is the hydrogen squenger. Let's say you want to produce hydrogen, but the problem is that within after some time the hydrogen squenger develop by default there unless you provide some pre-treatment. So they can eat, they can remove hydrogen. So instead of hydrogen, you end up with something else, maybe CO2 or maybe methane. So we have solved this technique also, this problem also. Uh, and another problem is to maintain high surface area, because we need high surface area of anode and cathode materials. As you can, if we have high surface area, we could increase the efficiency of the system. So we develop reactor with the novel design using some catalyst, also using novel type of electrodes, and we also quantify the internal resistance associated with materials such as anode, such as cathode, such as like solution substrate. We have quantified, and then we designed based of our model. So we were able to design very efficient system. So here you can see that, so we have developed basically novel hybrid electrochemical system. So what is that? As you can see here, this is the we use lignocellulosic materials, means the waste from agriculture waste, and wastewater we got from brewery industries. So here you can see that actually the anaerobic, this system is FMEC, fermentation as well as microbial electrochemical technology in one state, in one single reactor. This is a membraneless system. So we developed. So here the problem with alone anaerobic system, if we use like to treat wastewater, our anaerobic system is not a complete system. Is a some is only able to treat about 50 to 60%, and the end product of anaerobic system we cannot use. It's just like a waste, but the end product of anaerobic system is a really good substrate for microbial electrochemical system. So by that we could increase the overall efficiency, overall yield of hydrogen, because some of the hydrogen we can get from anaerobic, and some of the hydrogen from the end product of anaerobic fermentation we could generate from microbial electrochemical system. So here you can see that in this picture. So the lower part, the is the anaerobic fermentation. So anaerobic fermentation, anaerobic bacteria, they degrade organic matter and generate volatile fatty acids, which is VFA, which is the suitable substrate for microbial electrochemical technology, as well as it generates some hydrogen. Here you can see the reaction that from anaerobic, the theoretical, we could able to generate 4 mole of hydrogen, and from MEC, because it's the waste product we use from anaerobic fermentation in one single stage system. So from MEC, we could able to generate around 8 mole. So total 12 mole of hydrogen we could extract. This is the novelty of this system. Nobody developed like that. So we were able to perform the experiment. We were able to generate around 20 liters reactor. Later on, I will show. But before that, we have quantified. Before that, we have done some experiments at lab scale, and we were able to perform the experiment in that scale. Yeah. And here, so we start first. As I mentioned earlier, the most of the system is suffering from high internal resistance. So what we have done, we have quantified based on our model. We design system which has less internal resistance associated with its materials, and then uh, yeah. So here you can see that in the first figure illustrated that we have studied the electrode spacing. We have studied electrode spacing between the anode cathode. So there was spacing between 4 mm to 9 mm. So different spacing of electrode setup we used in the system. And with respect to buffer concentration, phosphate buffer concentration means this is the substrate for MEC. So as you can see, and with shaking and non-shaking in the first two graphs. So here, what we have done, so you can see we have found that with the spacing, when we increase space 4 mm to 7 mm to 9 mm, we found that there is not much increment of current density, because current density is directly, we basically measure the current density. Current density basically a flow of electric current per unit cross sectional area of the materials. So these electrons at the end, as I mentioned, it can be converted to hydrogen. So we can relate hydrogen with the current density. I'm not going to explain how this is the, I think is a basic thing. So the current density, so what we have done, we have studied different electrode spacings, and we found that with this range, the electrode spacing had not much impact on current density. As you can see that, but when the phosphate buffer concentration was 5 to 5 millimolar to 100 millimolar, the electrode density was linearly increased with spacing. But when we increase phosphate buffer is around 200 to 250 milligrams, so the impact on electrode spacing was not much, which means that is not much. But with the shaking, the electrode density was the bit high, which means that maybe the electrode density, the reason could be due to the uniform flow of liquid between the electrodes. So from here, we concluded that the electrode spacing is not much impacted the performance of MEC, but this result is somehow is contradictory with previous some published papers. So some of them claim that the less electrode spacing could increase the efficiency of MEC because of the transportation of the electron and proton. Is nearby. But even some of the study, they have reported that with the large electrode spacing between the anode and cathodes would limit the transformation of protons, which ultimately hindered the efficiency. So from here we concluded that the electrode spacing could be in this range. And further, we have also found that the ratio means anode to cathode surface area with shaking and non-shaking. And we found that what we have done, we increase first cathodes, number of cathode with respect to cathode anode. For example, one cathode in the system, we increase number of cathodes. So we found that in much not impact on the current density, as you can see in this figure. But later on, what we have done, we have increased number of anode So we found that this system is anode limiting. We need more anode with respect to cathode. The reason could be because we have used in the cathode side, we have used some type of catalyst, means like molybdenum phosphide. I'm not going to explain how we synthesize, because we have got this catalyst from our collaborator, and we simply paint on the cathodes. So which means that we make cathode more efficient. So we found that the this reactor was cathode sorry, anode limiting. So we increase the number of anode. We did not increase we do not increase any cathode, because as you can see that 4:1. So and we found that the shaking around 90 rpm with the cathode 4:1 ratio increase the electrodness. The shaking, the reason could be the, the dynamic flow of liquid between the electrodes. So the equal distribution of chemical and substrate to the bacteria, that could be the reason. So as I mentioned that, further we found that, as I mentioned that, there is many hydrogen scavengers exist in the reactor. So this is basically here we have done lot of work on this part, but I'm just explaining in brief what we have done. uh, the main the another reason of microbial electrochemical technology is suffering is hydrogen scavengers, because when we run reactor with any type of substrate, we end up with methane or some other biogas instead of hydrogen, because many like methanogens, homoacetogens. So we are the first one who found homoacetogens in microbial electrochemical, and we are the first one who developed this technique to inhibit homoacetogens and methanogens in the reactors. So what we found, we run the reactor with and without treatment of the culture. So treatment, we used chloroform. I know chloroform is not environmental friendly chemical, but we still working on that to the degradation of chloroform further into some other products like dechlorination. And the chloroform concentration we optimized around 0.01 ml. We used approximately 48 hours for 20-liter reactor, which is very less. And we found that at the end of the experiment, we checked the concentration of chloroform. So because some of the bacteria exist in this reactor able to degrade chloroform, 98% chloroform removed from the reactor. So okay, so we have given the treatment with culture with chloroform and without chloroform to justify the homoacetogens. Homoacetogen means when we start reactor, as I mentioned that for microbial MEC reactor, the acetate and volatile fatty acids is the best substrate. So here you can see in the first graph, when we run the reactor with the treatment, sorry, without treatment, the acetate concentration was, you can see the without treatment acetate concentration was almost like 40 millimolar, almost stable. But it should not be like that. The acetate concentration should decrease, because the acetate is the substrate for exoelectrons to convert into valuable product. So here, but when we run the reactor with the treatment of chloroform, you can see the acetate concentration was decreased, which means that the acetate was converting into hydrogen. So here, because if you see the anaerobic cycle, when we have acetate, the exoelectrons, they convert acetate into carbon dioxide and H2. But some acetogens bacteria, they again convert CO2 and H2 again back to acetate. So that is homoacetogen. So we able to inhibit that method, that the problem homoacetogen in the reactor. So with the treatment, you can see the hydrogen production was significantly increased, but without treatment, the hydrogen production was decreased. So and similarly, the as you can see that the current density of our reactor with and without treatment was stable, which means that, because I mentioned that the current density means the production of electron, not hydrogen. So the stable, which means that the electron were performing well for electron production. But when we come to hydrogen energy, the hydrogen production was decreased without treatment, because the exoelectron, they started homoestrogen, they started eating carbon dioxide and H2 to back to acetate. So we solved this method using chloroform. There is another, we have published some high impact papers on this. maybe you can see later on. So this is the mechanism. We found that to inhibit the homoacetogens, there is a good jungle pathway. So as you can see here, the hydrogen and CO2, it comes from organic matter, or from when volatile fatty acid like acetate, butyric acid consumed by the bacteria, they generate H2 and CO2. But again, the acetyl coenzyme, some homoacetogens, they consume again H2 and CO2, and again they produce acetate. So here we solved this method. We use some chloroform. We stop the homoacetogens and be able to enhance the hydrogen recovery, energy recovery, as well as, of course, the wastewater treatment is there in the anodic chamber. So this method we are working on now to scale up, to use for large reactors. Maybe we have already optimized for 20-liter reactors. So the next target maybe up to 5 to 100 to 200-liter reactors. So further, again another problem I mentioned in the starting, the quantification of internal resistance. As I mentioned that most of the microbial electrochemical reactors is suffering from internal resistance, which is associated with their materials, anode, cathode, solution. So we have developed the model. So by this model, we can quantify internal resistance, and we can tell the people, and to quantify, we can tell the people to design based on this model to their reactors. So here we have run the reactor by applying different voltage, as you can see that between shaking and non-shaking, and we have found that with the shaking, the internal resistance was less in terms of with respect to solutions, which means that because equal distribution of the chemicals between the electrodes, anode and cathode. So here we have predicted the shaking and non-shaking, the internal resistance we have predicted, we have compared with the observed and the predicted, and the result was pretty much similar. As you can see here, we have found the non-shaking and shaking, the difference between internal resistance, and we have quantified with the non-shaking the anodic resistance was 210 ohms, as you can see here, with shaking 150. Similarly cathode, similarly with the solution resistance. So yeah, as you can see that balance anode to cathode surface area, as I mentioned earlier, we have optimized the ratio. So for the shaking 3.5 into 1, and we found that with the shaking, using this model, we quantify the internal resistance associated with anode, we quantified the internal resistance associated with cathode, we quantify the internal resistance associated with the solution, and we optimized. So from here, as you can see that we optimize that buffer solution, with shaking, the optimum, we found that the internal resistance with 200 millimolar buffer solution, we found that internal resistance 57 ohm, but with shaking 27 ohm only, which means that we can use less buffer concentration for our MEC rather than feeding with high concentration of MEC. So we could save cost. One point. Another point, we can design our electrode based on the surface area, because if we know the internal resistance, so based on this, we can easily generate. So another, that the effect of shaking on electrode resistance might be why we have less resistance with the shaking, due to the influence of local chemicals concentration change at electrode fluid interface and proton and substrate consumption. So these results also suggested that, so what we have done, we have used this model to predict some other studies, which is not done by us. We can predict other model, we can predict other designs, whether they have designed their reactor efficiently or not. So the result suggested that the specific resistance of the electrode and solution can be used to predict the performance of MEC, and not only in this study, we can predict somebody else study also, and we can tell them, okay, you design according to these conditions. So here we have designed, like Liang et al, Chang et al, we have predicted their study, and we found that the in MEC found a high mixing condition, the other is low. we found that the Liang et al. Was suffering from the high mixing condition because they do not use the mixing condition. So that's why their resistance was high. So we can easily predict. Another study, Chang and Logan, we found that most of their reactor, the solution and cathode resistance by solution resistance. So which means that you using this model, we predict and we tell them the resistance in your reactor is due to the cathode and solution. So the highlights, the main highlights, main finding of this work is we have developed, quantify the model, and we have also able to balance the internal resistance, which is distributed with different materials, so we can make reactor. So based on that, we have developed reactor. As you can see that around 10 liter, but we have 20-liter reactor also. So this is the upflow hybrid MEC reactor. I already explained their construction. This is the high width longitudinal structure. So here we have used different setup, as I mentioned earlier, the 3:1. So we have different setup. As you can see here, this is the 3 electrode setup. This is the 5 electrode setup. Here anodes, like 3 anodes, 1 cathode. Similarly, with different configuration, we have different type of reactor. So based on that, we found that we have run the reactor. This is a 20-liter. Again, we have different setup of reactor, like 3 electrode combination, 5 electrode combination. Based on that, we have run reactor with brewery wastewater. As you can see here, I have a video also, but I don't know how to show here. So as you can see the result, we were able to generate around 6 liter per liter per day hydrogen from this reactor using brewery industry wastewater up to more than 50 hours. So this reactor we run more than 6 months with the stable performance. And with synthetic substrate such as phosphate buffers, we were able to generate more than 12 liter, which was very close what we have predicted earlier, less than $2 cost. So we are still working on their further, like for utilization of CO2 and in this reactor. So we have able to generate, as you can see that the current density with chloroform, without chloroform, the hydrogen production with the chloroform, you can see we able to generate almost 6 liter per hydrogen per day from the reactor. So yeah, so we have compared also our results, and we found the our reactor is the till date is the best as compared to other reported studies, because here we use very cost effective electrode material. We quantify the internal resistance, we design according to that. The internal resistance was very less as compared to the other. That's why the performance was higher as compared to other reported studies. So further, we are working also, as I mentioned earlier, the food productions, and still our work is going on. Utilization of CO2, conversion of CO2 and wastewater into energy, as well as animal feed, aquaculture. So this is our future work, not actually future work, ongoing work. We have already started working on some type of single cells protein production, aquaculture meal production using this technology together with energy production, air, and as well as wastewater treatment. This is the overview of current and future works. So the summary, uh, yeah, summary of this work: the cathodic hydrogen recovery may decrease under the long term operation. Like, let's say if you want to run for 1 year, maybe the hydrogen production recovery is decreased due to the again growth of homoacetogens, but we have found the method to inhibit homoacetogens. Another, we should also work on low cost materials for electrode and at cathode, although we have used carbon cloth, which is very cost effective as compared to the precious metal like electrodes, platinum, and some other. So yeah, we have developed model to quantify the internal resistance. So further, the output of this work we have published. Maybe if you want to learn more, you can go through publication. We have published quite high number of papers from high impact paper like chemical engineering around 8, and trends in biotechnology 13, environmental international related to specifically related to this work. So this is the thanks to my students and also researchers. These are my students where I work. They have done already PhD and working in all around the world, and some undergrad students. So for additional information, maybe related to this work, you can go Google Scholars and my publications. And we have also published a few books recently, like related to this technology, from American Chemical Society, CRC, Springer. So for the more detail, you can go through, or let me know if you need any details. So thank you for listening, and please let me know if you have any questions, or uh, I'm glad to answer. Thank you. Yeah, thank you, I'm done. Hello.
Okay, do we have any questions? Like I'll display the questions. Okay. Any questions? If anybody wish to ask any questions, please ask. Can ask.
Yeah, please ask if you have.
Am I audible?
Yes, yes.
Thank,
Yes, sir,
thank you,
you're
Professor
audible.
Lakshmi Singh, for a
Yeah,
wonderful presentation.
thank you, thank you.
We appreciate that, for sustainability, food, water, and energy, all the three are very important. And as
Yeah,
you said, we need 60% more food, 10% more
yeah,
water, and higher
yeah,
energy. So it was very
yeah,
exciting to see agricultural sciences has already made adequate advancements, and
yeah,
we have a projection that by 2050 we be able to meet the food requirement. But water is one of the major limiting factor in agricultural production. Your projection says that 10% more water, and water availability is going down day by day. If I remember the statistics for India, in 1950, we had 5,000 cubic meter of water per capita per year. That was the fresh water availability, which became 1,500 cubic meter per capita per year
uh-huh.
in 2010. And by 2013, we are expecting it's going to be 1,000 cubic meter per capita per year. So you see, in 50 years, the fresh water availability per capita in India has gone
Yeah,
down by two-thirds, from 5,000 to 1,500.
uh-huh.
And from 1,500, it's going to be 1,000 cubic meter per capita per year. So, how to meet this requirement? Your proposal for wastewater treatment is very, very attractive. And
Mm-hmm.
from wastewater, you have shown that not only energy, but also we can get the water which can be recycled for irrigation.
Yes, yes.
So it was really a wonderful presentation, and from sustainability point of view, it's very, very important. On behalf
Mm-hmm.
of the society and on the behalf of the president and general secretary, we would like to congratulate you for wonderful presentation and your having spared your valuable time to share your experience and research on this. Thank you.
Dr. Lakbir, a question is coming. Dr. Lakbir,
Yeah,
a question is coming. What is the
yeah,
fate
sure.
of byproducts in the microbial fuel cell? I
Yeah,
repeat,
as I
what is the fate of byproducts in microbial fuel cell?
Yeah, as I mentioned that we can extract number of valuable products, but again, it depends on bacteria and it depends on substrate, what type of substrate you want to use. So currently, like I mentioned, we are working on in one single system where we can use lignocellulosic biorefinery CO2 and brewery industry wastewater. So we were trying to convert that CO2 into single cell protein and for energy production in one system, as well as we reclaimed clean water and some nutrients recovery from the wastewater. So number of product can be generated, but again it depends on your for example your objectives of the work. Let's say your focus is only methane, then you have to use some methanogens. Let's say your focus is on H2O2 production, then you have to play with your bacteria. So but this system can be used for number of chemical production from any type of biodegradable system.
Okay, one more
So,
question is hitting. Can
yeah.
we produce oxygen? Can we produce oxygen?
uh Oxygen, I think we do not need to produce oxygen, right? It's already in the environment. So
Environment.
yeah, so oxygen. I didn't see particular on oxygen, but but we can produce number of other valuable products and biogases. Yeah.
Yeah, another question is hitting. Why hydrogen electrolyzers are designed to round stacks instead of the square one like fuel cells?
Can anyone explain me why hydrogen electrolyzer are designed to round stack? Electrolyzer to round stack. Actually, this question I think is from fuel cells. We do not need any hydrogen to synthesize to like round neck. What we have done, we have designed a reactor in simple way. We have designed the reactor in longitudinal structure, and what we have done to purify the produced hydrogen, we could use some type of membrane which we can separate from produced biogas. It's very simple design for microbial fuel cell. But like fuel cells, we did not design in some specific shape. We just use some type of membrane in the top of the reactor where we can easily separate hydrogen from other biogas.
Astha Yadav is asking what is the most effective methods of for storing
Again,
hydrogen produced by MECs?
yeah, again, the hydrogen, the storage is another field of, I think, research. What we have done, we produce hydrogen and either we can directly use because the production rate was not much. But what we have done, we simply exposed to the environment because there was no facility for the storage. But many people are working on like nanomaterials, fluorine, what we called magazines, graphene for storage of hydrogen. So, but we simply store a little bit on the pressurized cylinder and then we use since we don't have much hydrogen production rate from the biosystem. But of course, for the long term application, we need the proper storage system. Yeah, and there's a very interesting field also to store because it's the major problem with the storage.
One more question is coming for you. Will
Yeah.
aluminum electrodes, cathode or anode, corrode when biased in pure deionized water?
Will aluminium electrode, cathode or anode corrode when biased in pure deionized water? Yeah, the corrosion is the problem, but is the problem when we place electron for like more than like one year or 6 months inside the reactor. So that's why we have to choose the electrode, we have to choose the material, we have to coat with some there is a efficient catalyst to protect the corrosion. Could be the chances are there. Yeah. So that's why we use carbon
Can
cloth and in the titanium frame we use carbon cloth, so there is no chance for corrosion cloth.
can bioethanol be produced by microbial
Yeah.
fuel cell methods?
Yes, why not? I mentioned bioethanol, methanol can be produced. A number of products can be produced. Fatty acids can be produced. Ammonia can be produced. There is number of products. H2O2 can be produced.
Okay, thank you very
Yeah,
much, Dr. Latvir Singh, sir.
yeah,
It was a very intellectual session, and I hope
yeah.
it worked for all our viewers. Thank you very much, sir. Now,
Thank
I
you
would
for
like
listening.
to invite Dr. Abdul Shokar bin Abdrazak from Malaysia. He is the MD at Managing Director at UMP EcoPest SDN, Faculty of CET University Malaysia Pahang, Malaysia. Dr. Abdul would be speaking on the potential of green technology solution in preserving the ecosystem. Handing over the mic to Dr. Shokar.
Uh,
Yes, sir.
Hello. Okay. All right. Assalamualaikum and very good morning to all of you. Okay. I hope my title today I will share to all of you regarding my experience towards using the green technology solution for better life, yeah, for our environment. Okay. Are you seeing my slide?
No, no slides are not visible. Uh, you please share the screen and then open your PowerPoint slide. Yeah, it is visible. No, it's not visible. Can you please just re-switch it on? Okay, now visible. Here. You can
Is
start
it
your PowerPoint
visible?
directly. Yeah, yeah, the PowerPoint. Okay, the potential of green technology solution in preserving ecosystem. Fine. Yeah, it's
Right.
there.
Okay. Okay. Thank you very much, Mr. Chairman. Okay, I would like to share to all of you regarding my experience towards using a green technology solution in preserving the ecosystem. First of all, I would like to inform all of you that this is based on my experience to using three of my green technology product called Phytogreen, Pisoptera, Pescilla, and Wasra. So before that, I will share to all of you what is the spirit to make sure that I'm using the green technology solution to make sure that it can be commercialized. It's not only for what call for personal, but also for our community at large. So before that, I would like to introduce myself first. My name is Abdul Shukur bin Abdul Razak, a senior lecturer from Faculty of Civil Engineering Technology, University Malaysia Pahang. And since I also become a managing director for the spin-off company. It's called UMP Ecopest Sdn Berhad, one of the spin-off company under University Malaysia Pahang. And also I'm one of the director at the East Coast region, Kuala Lumpur International Chamber of Commerce. So, and treasurer and international council of SME and entrepreneurship Malaysia. So before that, what is the spirit of me to boost the green technological solution as a medium to protect our environment is our philosophy. Our philosophy and the core values of university is most important. The five core values University Malaysia Pahang, I bring it to make sure that I'm trying to realize that the technology, green technology, can be commercialized and also to protect. The most important is protect our environment, especially for our next generation. So I just brief about the 5 core values, the 5 important core values of UMP. It means give me the motivational encourage me to make sure that the green technology is most powerful to make sure that we can change. It's not change the world, but also we can change a small part and protect our environment. So it's called the strong bond with the creator and steadfast in upholding shared principle. It's most important. Everybody in the world have a belief to the God, and we must protect our mother nature to make sure that we not only for ourselves but also for the next generation as well. So then steadfast in upholding share principle, you must have a principle to make sure that whatever technology you want to use is beneficial to others. So and the third one is a creative in making wise decision. When we talking about the potential of green technology solution in preserving the ecosystem, and then we must have a creative, innovative to make a wise decision. And smart is most important to make sure that we have a specific, measurable, achievable writing and timeline. So and then this is what called a smart partnership. I show a figure smart partnership how the creativity and innovative will make sure that we can boost and show that the what medium or technology we want to use, we can make it a challenge and what called a change to not only at that area but also in the ecosystem. Okay, and then work smart, high impact. So when we talking new technology, especially when we talking about a green technology solution, when we must give the and work smart and high impact. And then we at this work smart high impact, we must have a strong teamwork, strong academic leadership, apprenticeship, outcome based, and the project must have outcome based. And all projects should be published in journal, postgraduate, undergraduate. Make sure to the technology is well known enough, and everybody know about this technology using to overcome the issue raised from that area. And then we must have a niche area of expertise. Okay. So what spirit of me is because research-based environment is a culture and is a tacit knowledge and structure, not only in the university but also by ourselves again happy interest, and then somehow is as a vacation and especially and to make sure that we can encourage motivation. We must have motivation to make sure that we realize the technology we want to Okay, discourse environment, publication for sure, and then new academic and research as a soul grouping, and will become high impact and to public and give the impact to the public and also for sure for our environment. And then the fourth core values is resolute in facing challenge. We must have face the challenge. If not, we not move. So we must take the challenge, especially now under MCO, under the outbreak and pandemic of COVID-19. We must have creative and innovative, as I mentioned before, as a core value number 3. So we must take the challenge to make sure that we can move from one point to another point and more better point to make sure that we can take this challenge. Okay, So this is my team to make sure that we can use the medium of green technology to make sure that we can overcome the issue arise in that area. And last is most important, proactive interaction action. So now when we talking about proactive interaction action, internalization, what others want from us, and then in this we can compare and expand our knowledge to not only in our region, not only in our territories, but also in the worldwide. We can share our knowledge, expand knowledge to others, and then followers and disciple and expanding experience toward global concerns, and then we can build network like this. International webinar is build a network, a friendship, and then as most important, branding conscious and holiday destination. Maybe not this time, but maybe for the next time. And then after MCO, the outbreak of a pandemic COVID-19 is okay, stable, so we can build network extensively, and also at the same time we can holiday. So and then academic interaction like this, okay, knowledge of common interest, negotiation skill, synergetic personality, communication knowledge, focus on research, publication, sharing idea, career path, international scholar, enable and enjoy traveling and holiday abroad. This is, I'm trying to show and to share to all of you how I'm trying to promote the green technology solution for preserving our environment. So, and then as academic, as a what called a businessman, we can we can teach, or we can become a mentor to serve the university, to serve the community, to serve the state and our country, and then to discover, publish, to tell the truth, and to reach beyond the world, and to change, to make the change. This is the most important. I'm trying to encourage myself and others and all the communities out there to make the change using the green technology, using the green technology solution for any what called issue arise from this world. So because of that, so we can protect Mother Nature. Now our the global issue, global warming, and the disruption of habitat, and it becomes worse. So as an environmentalist, we have a human being, and then we must have a green-minded. So using the green technology solution first, before we using others chemical or to overcome the issue raised up, the problem or the issue raised up from this world. Okay. So and then so we must have a world-class academic. We must have a world-class researcher, and then what criteria and benchmarking and trade to be this. And then innovative specialist area. Okay, so I'm sure that Prof. Lakve just now is an expert in the microfuel cell. Okay, and then this is you must have a specialist area in what you involve. So for me, I'm the environmentalist. So I'm trying to promote the green technology solution is a powerful solution to overcome the issue or the problem raised up from our area. So sufficient funding for research and networking is for sure. So we must have a sufficient fund to make sure that our research is done properly and smoothly. So commitment, passion for knowledge, leadership, mentor, motivator for academic and research, academic career and development. So as I mentioned to you, and then when we do the research and development and commercialization, this knowledge from as an academic field and research journey are a marathon. A marathon, even that are colorful, long process, and yet enjoyable experience. So money will come later. So you must show your best performance in what field in the field are you expert, especially for me. I'm trying to show that the green technology solution is a powerful tool to make sure that we can overcome what the problem or the issue raised from this work. Okay, so this is and then as I mentioned earlier, just the when we show your passion in research and development, when we sincere to do the what you call that work is sincerely, and then you for sure this is will come later. I want to show you how green technology solution can overcome the issue raised up in that area in your region. So this is, I'll share you about 3 or 4 of my research product related to green technology. So this is called PhytoGreen. So I'm trying to show that the development of domestic wastewater treatment facility using integrated of bioremediation and phytoremediation technology for refurbishment of oxidation pond process via PhytoGreen UMP. So PhytoGreen is the end product from the green technology using the phytoremediation and bioremediation technology. So this is about this actually this wastewater treatment is already there. It's about 30 years ago. So I'm trying to refurbish and to upgrade this pond using the green technology called phytoremediation. So this pond I'm being is about what called is about it is about 2 times of football field. Okay, so I'm trying in the oxidation pond we have 2. Okay, it's called facultative pond, and the other one is maturation pond. I'm just manipulate only for maturation pond, so to make sure that we can produce more better effluent quality at the end of before discharge to the stream. Before that, I just want to ask the secretary how long are the presentation? Is it 45 minutes? Hello, secretary. I just want.
You can have 10 more minutes.
Oh, 10 more minutes. Okay. All right. Okay. So this is about the green technology. Okay. And then I'm proof that this is the first effective technology, green technology in Malaysia using the phytoremediation technology. So this is I'm using the aquatic plant called Gladiagas in local name. Okay, this is a water lettuce. Okay, this is what called PhytoGreen in news. At the time we got the coverage at the national Malaysia. So this product, I'm sure to you that this product is when we want to commercialize, we must have what called IP, intellectual property. So I patent this product as a process for treating wastewater, and the other one I patent the system that's called a floating apparatus for cultivating aquatic plant. This is I also patent the trademark called PhytoGreen. Okay, so this is recognized in national and international level. This is just a short brief. You can see this is before and after. This is before the green technology solution called phyto remediation take part. Okay, you can see the on the surface of a maturation pond, all and grease and scums there. And then this is the what called effluent discharge to the stream uh, because the pond is already dead at the time. So, so you can see after using the green technology solution, in this case, we call phytogreen, using the phytoremediation technology, you see like a mirror, the lake is become beautiful. Okay, the oxidation pond at the maturation pond is become clear and shining. Okay, so you can see before and after. So before is a dark color in black, and the other one is a clear color. And the after 6 months of the duration. So why this happen? Because phytogreen or the plant itself is you have a natural membrane. So this is under scanning electron microscopic. It shows that cattle or Typha angustifolia, Lepronia articulata, Linocane flava have a natural membrane and is not it can't see using the organoleptic our eye, but this can see under scanning electron microscope. And then if I have a time, okay, I want to share this is a second product. So I'm using the empty fruit bunch as an environmental friendly pesticide to overcome the issue or the problem come from termites. So we using the waste from fresh fruit bunch called empty fruit bunch as a medium for attract termite, and at the same time that empty fruit bunch is already treated and then to what all to make sure that the termite not at all at that area, or we can replace it or prevent the termite come to that premise. And there's at the time in 2006, my research in 2006, at that time Olympic what call sport, okay, in Beijing in 2006. So the termite attacked the Olympic what call complex, and then that complex is collapsed. So the government of Beijing at that time asked all the researchers to find out the cheaper solution to overcome the problem raised out from the termite. So this is this product only can give a service to soil treatment and also in the residential area to overcome this problem from termite. Okay, and then this is the latest. I got project in India, Hojai, India. So a big what called plantation of agar wood. And then Alhamdulillah, I show that this product have a potential to remove, to repel the termite from that area. Okay. Agar wood farm in I.G.B.R.D, Hojai, India. It's about 1,000 acre there, but the pilot study in I.G.B.R.D is about 1 acre. Okay, this is top 5 finalists, and at the end, the what called the motivation for me and my group is we can use the green technology to be commercialized, and at the end to make it it's become as job creator to others. We not seek for job, but we ready for job creator to the community. And then this is a piece of terra in news. Okay, termite bitten pulp tank. So we can repel, we can eliminate the termite. So at that time, so we got news lah. The product is published to Malaysia. And then the third product is WASRA, water supply in rural area. I'm using the activated carbon as a filter to remove all the contaminant from the raw water. Because if I share here, especially in the remote area, the Aboriginal community take water from the river, from the what called the tube well. It's not clean. It's not what called clean for for drinking water purpose. So you know the penetration of what called the penetration of fertilizer at the to the tube well. So and then the tube well become contaminated by fertilizer. So it's not good for their cell. So we must have a solution to make sure that we can protect their house, and then so we using the activated carbon from waste, from coconut waste, and also from the empty fruit also. So from the tagline is bringing the idea from urban to rural area. So we must make sure that the community at the urban area, the rural area or remote area must have get a clean water, same like as the community in the urban area. So this patent granted in Malaysia also. This is recognized by our previous Prime Minister, Datuk Najib last time. So Walsa in news, okay. So and then the latest achievement, and then we got recognition from our government to recognize this product is under IBO, Innovation Business Opportunity, and Alhamdulillah, it shows that when we using the right and the good green technology solution, is not only can solve a problem in certain area, but also in our country, in the world also. So when we serious to make sure that the green technology solution is a powerful alternative or solution to make sure that we can preserve our ecosystem. As this wastewater treatment using plant, so the potential revenue is about 2 million. Okay, but the piece of terra potential revenue is 14.2 million. It's not audited by me, but this what called this survey is from the Oxford University, give to our government to see how potential the green technology solution to overcome the issue raised up in the world. Okay, there's 3 product commercialized already from my experience. Now I'm become a managing director of a spin-off company, University Malaysia Pahang. So become my position is managing director. I'm trying to prove. I want to share to all of you. Don't worry. No need to high-tech technology can solve the problem, but also when we use a simple but high impact of the technology, especially green technology solution, we can change the world. We can change the scenario of the complexity of the issue raised up in our country, but specifically, but also in the world. That's all for my presentation. Okay, if because have a limited time, actually, if you have any question want to ask me regarding the what
Yes,
called
Dr.
green
Shurkar.
technology solution.
We are hitting with a question from Astha Yadav. We have a question from Astha Yadav.
Yeah,
Can hydrogen fuel cells serve safely as green energy?
yeah. Thank
I
you very much,
repeat, can
Joe.
hydrogen fuel cells serve safely as green energy?
Yes, thank you very much for that. Can, because the hydrogen fuels can produce from the waste, especially chicken waste, fish waste from the wet market. Based on my experience, I'm proof already using the waste from wet market. This is called from cat cat from the catfish and also in the chicken waste from it shows that have the hydrogen fuel can we can call a green technology solution because we can produce it from the organic waste like chicken waste, catfish, wet market. And from my experience, it can produce alternative energy. Electricity is about 1 kg and produce about, if I'm not mistaken, 0.5 volt. So if we use fully utilize all the waste from the wet market, so in the big scale, I'm sure that so we can, especially we can remove, we can control the what called organic waste enter the stream, especially our river. So because we reuse it to become alternative energy using the microfuel cell. I'm sure the hydrogen fuel to overcome the problem raised from the wet market. So meaning that when we reuse the waste from wet market, or we can call it from the organic waste. So it's not only to produce alternative energy, but also at the same time can become a very good organic fertilizer for our plant, our crop plantation. Yeah, can, no problem.
Uh, another question is hitting. Can change of perception change the intention of to use a non-green technology in environment-friendly way?
None. Can you repeat again?
Yeah. Can change of perception change the intention to use a non-green technology in environment-friendly way? You can also see it on your screen. It's visible on the screen as well. Dr. Shokar, am I audible?
Green technology be used in environment friendly way? That's the question. Dr. Shukar, I think my mic is not working.
I guess he is not able to listen us. No issues.
Listen us.
Okay, I hope someone else can answer to the question. Uh, okay, he left. He left. Okay, no issues.
Okay.
So now I would request Professor Behel, Professor R.K. Behel, who is our next speaker. Professor R.K. Behel, sir. Professor Behel, can you please unmute your mic? Professor Behel, can you please unmute your mic?
Michael. Hello.
Yeah, sir. Okay.
Uh,
So I would like to introduce to our next lead speaker, Professor R.K. Behel, sir. Professor Behel is the Dean, CCS, HAU, Chaudhry Charan Singh Haryana Agricultural University at Hisar, Haryana, and is currently the Secretary of SASARM, India. Professor Behel would be speaking on challenges in food and health. I warmly welcome you, sir, and hand over the mic to Professor Behel.
thank you, Ashman. Yeah, first of all, I uh, thank the organizers for giving me the opportunity to talk to the international community of scientists and fellow participants from India and elsewhere. My topic is a very general nature topic, and every day in every house we discuss about food and health. Food is any commodity which we consume to satisfy our hunger and to meet our nutritional needs, energy needs for body functions. Now, food availability, food affordability, food accessibility, and food social acceptability determines whether a food is worth a food and is accessible to the human beings for consumption. Enough food is there in the world, but then the supply chain is still questionable. Therefore, that causes the problem of food supply chain management. There are some people who are hungry, there are some people who are overfed. Hungry people, less fed people, overfed people all have the health issues because of the food problems. Around 915 million people around the world, they are suffering from malnutrition. Around 2 billion people are having the mineral nutritional deficiency, and the hunger deaths are not uncommon in many developing countries. So that is the food scenario. When it comes to the essential quality of food, it has to provide adequate nutrition. And what are the basic principles through which it is provided? There are mainly six ingredients in a food or in a healthy food. One is carbohydrates that meets the energy requirements of the human beings through our food. They are also known as caloric food. Wheat and rice are mainly contributing to this category of foods. Then we have lipids, fats. Then these are very essential for maintaining the locomotion of the joints, packing of the muscles, carrying out many reactions in the body, metabolic reactions. And in this category, the important crops are brassica, groundnut, maize oil, rice bran oil, then peanut oil. So these are main categories of the oils which meets our lipid needs. Then the next category of ingredient is proteins. Proteins are very much needed for buildup of cells, tissues, organs, muscles particularly, and these are provided by mainly by legumes, chickpea, urd bean, mung bean, cajanus cajan. These are the principal crops which are providing the protein needs. Then we have vitamins which are provided mainly by fruits and vegetables. Vitamins are very much essential for carrying out the physiological functioning of the body, metabolic functions of the body. They maintain the growth rate of the body, they are essential for the mental health, they are essential for the, the metabolic reactions, and these are provided by the fruits. Then we have minerals. Minerals are very much deficit in Indian foods because neither the agriculturists put mineral fertilizers in the soil, and the soils are already poor in the mineral. Therefore, the mineral nutrition through the major category of foods like wheat, like rice, that deficiency is not covered until unless we breed for harvest plus type of varieties to provide more quantities of mineral nutrition. Major mineral nutrients are iron, zinc, copper, manganese. They are needed for maintaining the electron transport chain in the body and the energy systems. The oxidative stresses are removed, and the muscular functions are also maintained by the mineral nutrition. Therefore, we need to supply enough mineral nutrients in the food. And fortunately, all the coarse meals like pearl millet, like maize, like ragi, they are rich in mineral nutrition, and therefore their integration in the diet is must. And we have another category of mineral rich foods, that is amaranth, in case of which is so far considered as wheat, but it's now considered as a reform food throughout Europe. And then there are spinach, which is very big source of mineral nutrition. So then, then we have vitamins E and C, particularly relevant in the pandemic times, because E and C are the vitamins which contribute directly to the immunity building. Immunity is required to fight out the corona pandemic, and Hippocrates said that let food be your medicine and medicine be your food. That is rightly said. If we can have the immunity building food, which is available in all category for all category of people, then it would be really good that we can ward off the damages by pandemics like corona, earlier also like we had the swine flu, we have influenza virus around. So that is also one of the essential way. And vitamin E is provided by fruits like papaya, garlic, which is accessible to all, superstar, then onion, consumed in every home, spinach, affordable by everyone. And then there is another category of mineral nutrition, that is bioactive compounds. All colored food products, fruits, they contribute to this kind of category of nutrients. Most of the colored fruits having red color, they are rich in anthocyanin, and anthocyanin is very potent antioxidant, and that is how it cleanses the free radicals from the body system, and it avoids diseases, initiation of diseases like cancer. So these are the principal foods, and water is the another category of food, because if water is clean, water is adequately rich in mineral matter, that is uh, that the TDS is neither very low nor very high, it will be good for the health. Now, what are the emerging issues in health? Now, the history of food is history of human being itself. The food is variable in different geographical areas, in different climatic conditions, but principally the food is vegetarian food or non-vegetarian. And these vegetarian foods, non-vegetarian foods, they carry their plus and negative impacts both. So foods not only provide energy, but their deficiency or excess of particular factor, they may lead to troubles, problems, also health problem, health issues, particularly in case of non-vegetarian diets. It has been seen that the protein content, excessive protein content, obtained through pulses and obtained through meat, they have different consequences. So the modern problems or health consequences are the infertility in women, impotence in men, deficiency of testosterone, deficiency of hormones, deficiency of minerals that leads to depression, that leads to anxiety, that leads to cancer, that leads to obesity. So these are some of the current health problems. And the each and every health problem can be tackled by consuming a particular diet, and that is the balanced nutrition. For obesity, it is generally said that consume less carbs, less oils, and consume more proteins. Protein has a compound, ghrelin, which quenches the hunger. And one person taking more protein eats less over the day. And that is the key to success, that one loses weight. Exercise is another sequel along with the diet for obesity problem. Then for the reproductive disorders, vitamin E is principal compound which should be consumed in right amounts, not less, not high, because if the estrogen production producing foods are consumed in higher quantities, there is again a danger of breast cancer in women. But then the optimum level of vitamin E has to be consumed, and that can be provided by legumes, by the vegetables, very cheap vegetables, by nuts for the rich people, by fruits, and by the Ayurvedic uh, preparations of bhasmas and various other herbal medicines. Like that, for reproductive health, again, the good amount of vitamins, vitamin D is immunity builder, and vitamin D is provided by mushrooms. And then there are other cereal, coarse cereals, which can provide vitamin D. This is very essential for immunity building and avoiding many reproductive disorders in women. So vitamin D has to be consumed properly, and the vitamin D sources are the mushrooms consumed, the sunlight is the biggest carrier, but calcium can only be assimilated in bones in the presence of D3 vitamin. Otherwise, calcium provided by various foods like milk, by nuts, by grains is not as accumulated in bones till the D3 vitamin is adequately there. So the purpose of this message is that the agriculture by itself has to be improved in favor of the nutritional quality. So far, we have been doing the agriculture for caloric needs, but of late, in next last 20, 30 years, it has been realized that our foods are not delivering the proper amounts of nutrition needed for well-being of the human beings. Therefore, now the agriculture programs like Harvest Plus are targeted on increasing the quantity of iron, zinc, copper, manganese, and some vitamins. Triticale is one man-made cereal which has broken the inverse relationship between protein and lysine. Protein is needed for the muscle buildup, and lysine is very much needed for the functioning of brain. So this is one very good cereal. Another unconventional cereal is quinoa, from introduced from Australia and India. Quinoa is low glycemic food. It is digestible slowly, so that the satiety feeling is there once the quinoa is, and it is also low in gluten. It is very rich in protein. Therefore, the people suffering from celiac diseases, particularly children, they can consume this new food and can remain healthy. Like that, there are another traditional foods which have been forgotten. Okay, we have ethnic foods in each locality. They are produced in particular climates, and they are valuable for their food and medicinal purposes. And these foods are basically the foods produced naturally in deserts, in gardens. So these foods, vegetables should also be reintegrated into our meals once again. The Bajra is one example, very good example, that it is being now reintegrated in our daily food by the non-Bajra eaters. The ragi is another one good food eaten in southern India and Bihar, but now it is adapted by the diabetic patients in north. So these are ethnic foods. So these should also be consumed. There are several foods which can create problems. The one of the pulses, they have the alkaloids causing elephant foot disease. These pulses needs to be processed. Lathyrus is the name of that pulse. It needs to be processed before it is consumed, so that its bad alkaloid, can be removed, so that it does not cause the elephant foot problem. Like that, there are many oil seeds like flax, which provides much more quantity of polyunsaturated fatty acids for the heart health of the heart. It keeps the cholesterol low, keeps the triglycerides low, and perhaps this is the best medicine all over the globe for the heart patients. And then we should also avoid use of fats when they are overcooked, because the saturated fats become trans fats, and trans fats are very dangerous. There are many countries where there is a zero tolerance to trans fats. Australia is one of them. Japan is again another good country. And the trans fats are produced when the saturated fats, which we consume normally in our diet every day, they are overcooked or recooked. Then they become trans fats, and trans fats gets deposited in the heart and blocks the arteries. Therefore, the use of food, cooking of food, should also be changed. Broccoli is a wonderful food, providing good amount of minerals as well as vitamins. But when it is cooked, overcooked, in traditional way, its value is very low. But if it is steamed only and consumed, then it's very good. So we need to have three dimensions of food. What is the food, its qualities, how it is cooked, who should consume it, who should not consume it. Diabetics, they should not consume certain fruits and they should consume others. Having high soluble sugars, having high glycemic index, these fruits are not good for the diabetics, but for the energy deficit people, These fruits are very good, providing more soluble sugars for the quick instant energy. Therefore, we need to understand that what are the different dimensions, what foods are available in a particular locality so that they are available to everyone, whether they are accessible, whether they are affordable, whether they provide adequate nutrition, and how they are served, how they are cooked. These are few things which one should consider about food in relation to health. So, thank you very much. If there is any question, I can try to answer.
Uh, Thank you very much, Professor Bahal. Now, questions, if there are any questions.
Yeah.
Anshuman, can you take up the questions
There
from
are
the
any
invite,
questions?
invite questions from the audiences?
Yeah.
Yes, sir. We are asking questions.
Yes.
Professor Bahal's talk is open for discussion. And if there are any questions, he will be happy to reply. He-
So I think that these are the general issues.
Good, good. I think there is no question. The talk,
Yes. So,
there are no questions. Thank you,
so
Professor Bahal,
thank you, thank you.
for sparing your time and being with us.
Uh,
Okay, thank you. Okay.
Okay.
okay, sir. One question is there.
Well,
One question is there.
Yeah,
Okay,
okay.
sorry. One question is there.
yeah,
Okay, the question is from Radhika Agrawal. Sir, how
yeah.
does the COVID pandemic change the world's food requirement and food supply chain?
Yes, wonderful question. Now you see that the COVID uh, era foods have to be immunity building foods. And I have already told you that the immunity building foods are available in all categories of vegetable, grains, and fruits. So the supply chain will depend upon the geographical region where you are. If it is a horticultural region, you can consume all colored fruits. They are very high immunity building fruits. The blueberries, red berries, gooseberries, then the, the colored grapes. So these are available in horticultural rich area. They are very good for immunity building. Then the condiments and spices are also there, particularly in localities like Kerala, they are highly produced. In Northeast, they are highly produced. Madhya Pradesh, they are highly produced. So they can have garlic, they can have ginger, they can have clove, they can have cinnamon stick. So they can maintain their immunity building foods like that. And in northern part of the country where neither the condiments are produced in much quantity, grains are there. So QPM maize, quality protein maize, is one very good source for immunity building because it provides vitamins contributing to immunity. It provides protein, it provides carbohydrates, very good food. Triticale is another very good example. So the supply chain has to be managed locally and in view of what can contribute to the COVID-19 resistance. And we have several ethnic foods grown in the villages. They must be brought to the knowledge of the city dwellers and they should be produced because they are naturally produced. They are neglected. Their production can once again be taken up. Amaranth is one such food and can be supplied to the cities. So supply chain has to be redesigned. Supply chain has to be managed according to the pandemic areas. Our monoculture crops system has created problems, and because we mainly eat rice, wheat, sorghum in the southern part of our country, bajra in Rajasthan, Gujarat. But then these are monologue foods, and the food diversity from Indian plate is missing. So therefore, it's a very good question. We need to redesign this food supply chain in view of the corona times.
There is one more question,
Yeah.
Professor Bahal. It says,
Professor Behel, yes, sir.
uh, does
Yes.
malnutrition causes infertility in human? You said that malnutrition causes infertility in human. Does
Yeah,
it have
sure.
any effect on metagenesis?
Yes, you see that there are three important reproductive problems in human beings now realized on account of malnutrition in women, infertility, or preterm birth. In human beings, the testosterone quantity is decreasing, and therefore infertility is also increasing. And malnutrition has direct impact on the metabolic and physiological functions of the body, and particularly the hormone levels. And therefore, the reproductive health of the male and female is directly affected by the malnutrition, because they affect the malnutrition affects malnutrition for minerals, malnutrition for vitamins, malnutrition for enzymes. So malnutrition for the metalloenzymes, they are directly involved in production of biosynthates, leading to hormone production, and hormone production is directly related to the sexual fitness of the human beings.
Poonam
Like,
Mehrotra is
like
asking, sir.
malnutrition. Yeah, yeah.
Poonam Mehrotra is asking, Dr. Poonam Mehrotra is asking, does organic agriculture will be produce sufficient food during pandemic?
Not audible, not audible. Yeah.
Dr. Poonam Mehrotra is asking,
The question is not audible.
am I audible now?
Ah, yes.
Dr. Poonam Mehrotra is asking, does organic agriculture will be
Yeah.
produce sufficient food during pandemic?
Wonderful question, and thank you for asking this question. Organic foods are great in pandemic times. Why they are great? Because the nutritional aspect in inorganic and organic is same. That is nitrates for plant growth. They are nitrates in both ways. But in organic foods, in organic agriculture, the organic material gives rise to a lot of microbes in the soil, and they promote the uptake of zinc, iron, manganese, copper. So these metalloenzymes are produced in much higher quantity in organic food than in inorganic food. That's fundamental difference. Another is there are several vitamins which are available in higher quantity in case of organic food. And that is for these two reasons. The organic food is much better for well-being of the human beings than the inorganic food. Inorganic foods, they can also have adverse effect because of the heavy metals pass through the food chain in the food plate. All phosphatic fertilizers, they have some heavy metals like cadmium, nickel, they're coming from the splashes, different fertilizers. So in organic agriculture, these adversities are avoided automatically because the fertilizers are not added. All bioinoculants or biomaterials are added in the field. Bioinoculants include the bacteria, the fungi like mycorrhiza, which increases the phosphorus, and bacteria, which are pseudophoric in nature, they increase the contents of the mineral matter. So it's very good question that the organic food will be much better for well-being and for immunity building because of the higher content of vitamins and mineral matter.
Uh, Dr. Komal Kathuria is asking which Ayurvedic herbs are useful for treatment of corona.
The I will not contribute to it saying that it can be treated. No, this would be an overclaim. The corona can be mitigated, however, by Ayurvedic treatments. And if you see that the infection routes of corona, they either come through the nostrils or through throat, direct infection to the throat if the mouth is open and it makes an entry. Corona stays there for a while, maybe 3, 4 days. And all herbal items are very good for taking the corona out from the throat, pass it on to the stomach so that it disintegrates in the HCl present in the body. This is mitigation, not treatment. Corona becomes serious when the corona passes from throat to the lungs, where it elicits mucus, and mucus becomes hard, stone like, and blocks the respiratory tract. There we need ventilators. And no herb has been shown to break that kind of mucus stone like structure. So it is not the treatment, it is only the mitigation. And now another homeopathic drug, which is arsenic album, it's also not a drug for the corona. It builds immunity. So these both the Ayurvedic products, they are basically acting on cleansing account, keeping the throat clean, providing enough stimulus to the body organs to produce the secretions which will take corona out away from the consequential parts of the body, because corona then after lungs passes to kidney to liver, but the best is if it is thrown to the stomach in HCl so that it gets disintegrated. So mitigation is possible. The only treatment would be the vaccine, because corona has 7 genes. Corona produces different kinds of proteins, and the antibodies will be the answers for that. And we need to have a polyclonal antibody, which will have many kinds of antibodies in itself, so that to ward off the corona. So corona treatment is not feasible. That is overclaimed by homeopathy and Ayurveda. But mitigation is certainly possible. These are very useful interventions, much more useful than even the allopathic therapeutic treatments, because there are 3 steps in every disease: initiation, promotion, progression. And all Ayurvedic products, herbs, they are very much useful in curbing the initiation. So that is how they are indirectly involved in mitigation of corona. If corona is not there, it is prevented. So it is much better than treating the corona. So Ayurvedic herbs are very, very useful in prevention of corona. Thank you.
Thank you very much, Professor Behel. It was a nice session and we came to know about many new things. Thank you, Professor Behel.
Thank
Uh,
you.
Thank you.
Now we have Dr. Ajay Veer Singh sir, but as Dr. Shokar has also joined us, and we had
No.
a few questions left from Dr. Shokar. So I would request Dr. Shokar to please answer the questions which were left. We have a question from Asta Yadav. Can change of perception change the intention to use a non-green technology in environment friendly way?
Okay, about your question regarding is it perception, right? Regarding perception, change the intention to use a non-green technology in environment friendly way. Yes, sure. We can, as I mentioned earlier, we can integrate together between mechanical and non-mechanical. In my Pythogreen product, the combination, the integration of three-way aeration, phytoremediation zone, and then after that enter the aeration zone, and the other one after that we enter the clarification zone. So this is a non-green technology, but we using the mechanical part in the system. So to produce a better quality of effluent. So now in this treatment, wastewater treatment, I'm using the phytogreen technology, which then area is covered about 2/3 of the maturation pond, and it shows that the capability of aquatic plant to uptake contaminant from the domestic wastewater is workable. So the perception change the intention to use a non-green technology in environmental friendly way. Uh, yeah, but the most important is about education and environmental awareness must be what call to promote to make sure that everybody know about the green technology solution is a environmental friendly way to overcome the issue raised from our side. Yeah.
Thank you, Dr. Shukar.
Yeah. Thank you.
Any other question? Anshuman, is there any other question? Is crop water management a part of green technology?
Is crop waste management is part of green technologies. Yeah. Green the crop waste management is a part of green technology. Yeah. It's depend on how you operate it. If you from the crop waste management, you're using the what call green technology approach. It can call part of green technology solution. If you use in the crop waste management, you use more chemical, is not is can't call a green technology because you use chemical dosing to overcome issue or problem to that crop waste. So better you do the screening first to make sure that what is the best solution to make sure that the crop waste can reuse, can reduce or recycle 3R. So to make sure that other beneficial users can overcome the problem at that area. Any question anymore?
Okay, Anshuman, can we conclude it now? Dr. Upadhyay? Dr. Upadhyay? Can we conclude it now?
Yes, sir. Yes, sir.
Okay.
Sir, we can conclude, and I would request you to please conclude the session. Yes, ma'am. Ma'am, broadcast studio.
Hello. Can we go ahead?
Yes.
Yes.
Can I go ahead?
Okay. Yes, sir. Sir, you can go ahead.
With the, there is a cross connection somewhere.
Yeah.
Okay, am I visible, audible to
Yes,
all?
ma'am. Welcome, ma'am.
Yeah.
Welcome.
Good morning to everyone. Good morning to everyone. Good
Yeah.
morning, Dr. Gupta. How are you?
Good morning, Malvika. Good morning. Welcome to this session.
So good to
Oh,
see you, sir.
yeah,
So
yeah.
Sorry, I
nice to
was a
see
bit
you.
late because
And
I was given a time of 11:40, so
okay,
I wasn't ready actually.
I,
Uh,
I
so
think in
I
session
would
is
No problems.
40.
Okay.
I would
Okay,
request
thank you.
Dr. H.S. Gupta to please conclude the session so that we can start with the next session. Thank you.
Thank you very much, Anshuman. This session has been mainly dealing with the sustainability of food supply, sustainability of water availability, sustainability of energy availability. And Dr. Lakhveer Singh spoke on production of clean fuel like hydrogen from novel microbial electrodes. He showed very clearly that in by 2050 we would need 10% of more water for agriculture. But in contrast, what we find that water availability per capita, at least in India, world over and in India, is going down. To give an example to you, the water availability in 1950 was 5,000 cubic meter per capita per year in 1950, and it has come down to 1,500 cubic meter per capita per year in India. So in last 60 years, water availability per capita has come down by two-thirds. So from 5,000 cubic meter to 1,500. And in next 10 years, it's going to be 1,000 cubic meter per capita per year. Then now we have an increasing water requirement. Where should it come from? And the reply, what he, I mean, is comes from his presentation that wastewater treatment. And this wastewater treatment will not only produce clean energy like hydrogen, but will also be able to supply byproducts, and treated water can be used for irrigation. So that was the gist of his of Dr. Professor Lakhveer Singh's talk. Dr. Shokar dealt with the green technology through phytoremediation, and it's very much required again for sustainability that we use green technology. See, whatever natural resources we are using, we should try to see that you know the rate of consumption of natural resources should come down. If we use it with the speed we are using, I think these natural resources would not last very long. Be it a petroleum, be it fertilizers, be it soil fertility, be it water. I think we have to be really very careful about it. Professor Bahal, Dean from CS Chaudhary Charan Singh, Haryana Agriculture University, spoke about health food. See, we all know that we have come a long way from 1960s. World was facing a really famine during 60s, 70s, and today we produce enough food to feed the world population. Whatever problem we are seeing is seen because of the bad distribution. So if we really can take care of at present in distribution mechanism, hopefully we will be able to meet the requirement. And we are producing enough. The current concern is quantity wise we are producing enough, but quality wise I think we are not able to feed people with a nutritious food. And that is what Professor Bahel said, that almost 900 million people suffer from malnutrition. Almost 2 billion people around the world suffer from micronutrient malnutrition. So, what is required is two-pronged approach, whereas we keep on increasing our quantity of food production or food grain production, we must also make attempt to improve the quality of the food we give. And biofortification has come out to be a sustainable way of supply of micronutrients. Friends, I would like to conclude by saying, like, India, if you see the India's statistics, by 2050, we expect that we will have a population of 1.6 billion. And to feed 1.6 billion, we would need a food of around 320 million ton. We would need feed and seed around 80 million ton. So total requirement comes to around 400 million tons. This year, we are going to have around 280 million ton, 290 million ton food production. Now, to produce 400 million tons of food grain in 2050, if we see, currently we have around 40 plus million hectare irrigated area. So out of this 40 plus million hectare area, if we take 35 million hectare area under food grains, then we can, with the rate of 10 ton per hectare per year, 5 ton in Rabi, 5 ton in Kharif, we will have 350 million tons. Out of 70 million hectare area which is rain fed, if we again take out for food grain production 35 million hectare area, and with the 2 ton per hectare in rain fed areas, if we produce, we will be able to have around 70 million tons of food grains. So 350 from irrigated, 70 million from rain fed. We will be able to have around 420 million ton of food grain against the requirement of 400 million tons by 2050. So I think with the present level of technology, hopefully we will be able to feed. But the major concern is improving the quality of food, improving the level of micronutrients, level of protein availability. Today in India, around 70% of people are protein malnourished. So we need to supply more of a protein, more of a micronutrients, more of a high quality, lipids and fats, and more of a vitamins and minerals. And with the current trend in research funding, hopefully I think we will be able to biofortify or improve the availability of micronutrients, vitamins, minerals, and also the quantity of proteins. Friends, with these few words, I would like to thank the organizers, especially the president of the society, Professor Rajendra Kumar, and General Secretary Dr. P.K. Upadhyay, for having given me the opportunity to be with you and share my thoughts. It was really wonderful to listen to the three talks, and I would like to compliment all the three speakers and the president of the society and general secretary of the society for having organized a wonderful session in this symposium. And thank you very much. And it was a great pleasure to see Dr. Malvika Dadlani, and I'll be able to hear the next session presentations. Thank you very much. I will hand it over to Dr. Upadhyay, please.
Thank you very much.
Thank you very much, sir. Lot of thanks, sir. Uh,
I would like to extend my sincere thanks to Dr. H.S. Gupta, sir. Starting with the next technical session, I would like to invite Dr. Malvika Dadlani ma'am as the special guest and to chair this session, technical session 2. Dr. Dadlani is former Joint Director Research, Indian Agricultural Research Institute, ICAR, New Delhi. Dr. Dadlani is an eminent scientist of seed science and technology and has been honored with several awards with IARI Best Teacher Award, FAO Expert, Consultation on Quality Declared Seed, Bhumi Nirman Award. Ma'am is also the member of Executive Committee, SARC Seed Forum. Fellowship of several academic bodies, including NAS, and Honorary Research Fellow of Biodiversity International Sub-Regional Office for South Asia. We welcome you, ma'am. A huge round of applause on this virtual media for Dr. Dadlani.
Thank you. Anshuman, for your kind words of introduction. And at the outset, I must thank the organizers, Dr. Rajendra Kumar, Dr. P.K. Upadhyay, for giving me this opportunity to chair this particular session. Um, Please pardon me because I have not been introduced with the speakers, and that will be very kind of you, Anshuman, if you can
Yes, ma'am.
introduce the speakers for this session.
Sure. Sure. Starting with our next session, we have Dr. Agnihotra Singh sir. Sir
Okay.
is Scientist D and in charge COVID-19 testing, Department of Microbiology and Molecular Biology, ICMR, National Jalma Institute for Leprosy and Other Microbacterial Diseases, Ministry of Health and Family Welfare, Government of India. Sir will be
Okay.
presenting his lecture on epidemiology and human health implications of Microbacterium avium sub paratuberculosis in India. Handing over the mic to Dr. Agnihotra Singh.
Thank you for the introduction, Anshuman. And I welcome Dr. Ajay Singh for being the first speaker of this session. And we are eagerly waiting to listen to your presentation. Dr. Ajay Singh, please.
Yeah, thank you, madam. And first of all, I would like to convey my thanks to President Dr. Rajendra Kumar Sahab and Dr. P.K. Upadhyay Sahab and Ansuman for making all the things together for this very interesting webinar and giving me the opportunity to discuss about this epidemiology of Microbacterium paratuberculosis and its human implications. So I would like to share my screen. Uh, Anshuman, I think I have to share my screen.
Sir, you can click on share screen icon
Yes,
available.
yes, yes. But, it is showing you or not showing you right now?
No, no, sir. No, sir. Just click on share screen.
Yeah, after that, there is some window, entire screen or application window like that.
Entire screen, entire screen.
Entire screen, okay.
And yes, sir, it's visible now.
Visible now, okay.
You can open your PPT here. You can open PPT. Yeah, it's visible. Okay.
So, in my talk, I am going to discuss about the epidemiology and human health implications of Microbacterium subspecies paratuberculosis in India. And as being a biological students, yeah, these questions always come in our mind. What's the total number of bacteria on the earth? And the answer of these questions come from University of Georgia when Dr. William total estimated in 1998 that the total number of bacteria on the earth is 5 million trillion. This is the number of 5 followed by about 30 zeros. So we can say that the bacteria are our invisible housemates. They are everywhere, and the average, it has been estimated that the average human can swallow about 1 trillion bacteria every day, and as many as 100 trillion microbes live inside us. That is the number, that this is the number that is more than our total number of cells. So most of them are not harmful to us, and very few, about 2% of them are harmful to us and cause the disease and unwellness to the human bodies. So when we talk about the mycobacteria, then it has been reported that the mycobacteria causing more suffering than all bacteria put together on these planets. So this is the thing actually I would like to highlight, that the mycobacteria have a very significant pathogenesis and affect the human life. So this genus has about 190 species, and out of these only 50 are pathogenic to animals or human. Rest of the not cause any disease. But when we talk about the major human pathogens of mycobacteria, we have most of all about know about the TB and leprosy. These are the major human pathogen which cause the disease, as Mycobacterium tuberculosis, which is well known to cause the disease tuberculosis in human, and the leprosy is well known to cause by Mycobacterium leprae. These are the slides just to highlight the situation of tuberculosis in the world. And during the period of 2018, about 10 million cases, new cases have been reported, and 1.4 million deaths have been reported worldwide due to tuberculosis. Mycobacterium tuberculosis, that's why is considered as the most successful human pathogens in the world. And this is the global situation of the leprosy. We can see here about 2,08,619 cases has been reported, new cases has been reported from the world in leprosy. And the visible deformity has been reported about 6% of the cases. So the people who are thinking that the leprosy has gone, this is very hard to say that the leprosy has gone, because the new cases are coming. And and in any disease, when new cases are reported, it means that the transmission of the disease is on and the disease is there. So the more focused research is, and the management strategies are required to combat all these leprosy cases and control the disease in various countries, including India. This is the trends of leprosy in our country. We can see here a continuous line is there to show the decline of the cases in our country. But we achieved the elimination target in 2005 in case of leprosy. The elimination target means it is a number actually, that when the we report one case or less than one case in within the 10,000 population. So this target was decided by WHO, and India achieved this target, one less than one case per 2000, 10,000 in 2005. So after that, we still struggling with the disease, and the new cases are occurring at a stable number. So we are focusing on the transmission dynamics, and we would like to control the disease by managing the patients, early patients, and diagnosis and treatment basis. So this is the about the major mycobacterial animal pathogens. So when we talk about the mycobacterial animal pathogens, the two pathogens are there: Mycobacterium avium subspecies paratuberculosis, which is considered as the most successful animal pathogen, and Mycobacterium bovis, which is a well-known cause of bovine tuberculosis in animals. So Mycobacterium tuberculosis paratuberculosis belongs to Mycobacterium avium complex, which also include Mycobacterium avium, which is well known to cause avian tuberculosis. And this pathogen has about 94% genetic similarity with Mycobacterium avium and shared about 3,000 genes with Mycobacterium tuberculosis. It's caused jaundice disease or paratuberculosis in animals and has been associated with various diseases in human beings, including Crohn's disease and some other autoimmune diseases. So when we talk about the history of this disease, so Mycobacterium paratuberculosis was isolated in 1984 by John and Prothangam. These are the guys were the German scientists, and they isolate the bacteria from the cattle, and the disease is mainly affect the small intestine of the ruminants, of the animals. So presently there is a no treatment available, and the first cases of paratuberculosis was reported in India in 1918 in undivided Lahore. So we can see here these are the corrugations of the small intestine. This is the picture of a normal intestine, normal animal intestine, and this is the initial stage of the disease where the corrugations are just beginning, and this is the full-fledged clinical picture of a advanced stage of jaundice disease in animals. These pictures belongs to goats actually. These pictures are the clinical pictures of animals which have active disease. So we can see here the ribs are very much open to these animals, and the animals are very weak, and they have some very lean skin actually. So these pictures are can be seen at the roadside, and very much common in roadside. You can see here. So when we talk about the economic losses to jaundice disease, so the disease caused huge economic losses to the farmers and dairy industries, and in the forms of progressive weight loss, reduced milk production, lower slaughter value, and premature culling, and also reduced the fertility. These are some figures actually that has been reported the economic losses per year, and in different countries we can see here the US is bearing about 200 to 250 US million dollar losses per year due to this jaundice disease. However, in our country, in India, the national economic losses due to JD at national level has not been estimated. But very recently, in 2017, Dr. Krishna Datta Rawat has conducted a pilot study in Alwar, Rajasthan. And this study was based on some farmhouse which was maintained for the Holstein Friesian cows for the dairy purpose. And the herd prevalence of the disease was 68.5%. And he reported that around the losses occurred to this farm due to JD is around 54,444 rupees per cow per lactation. So we can estimate that the how much the disease affect the economy of farmers due to this significant effect on the reduced in the milk productions. So the transmission of JD in animals is mainly occurred due to the fecal oral route, and the infected animal excrete the abundant amount of bacteria in their feces and milk and semen. So the vertical and horizontal both type of transmission is there. So the young calf mainly receive the infectious dose from the fecal oral route from the infected animal or infected mother. Mycobacterium paratuberculosis has been isolated from various species, including domestic ruminants and wild ruminants, and including primates and human beings. So this is a very wild host range, and the diagnosis of the infection is very difficult. None of the single test can diagnose all the cases at a time. So it is always recommended that the use multiple tests to screen the disease in the herd. So, however, the culture of this culture is considered as the gold standard test to diagnose the Mycobacterium paratuberculosis. But it also has very limitations. Actually, the Mycobacterium paratuberculosis strains can be divided in three types. So, sheep types, cattle type, and bison type. On the these based on the mainly the molecular epidemiological test, and these strains have been reported to be profound on their own medias. So this is the one situation where whenever we go for the culture, so we just miss some of the strands, and the sensitivity of the test is also depends on the number of bacilli available in the feces, and subclinical infections, quality of Mycobacterium J, which is an ingredient of of this of their media, that is Herald's egg yolk media. So this Mycobacterium J is also required to promote the growth of Mycobacterium paratuberculosis, but the quality also affect the isolation rate of the bacteria, and quality of egg yolk has also affected. So there is a limited reproducibility, and the time consuming is also one of the limiting factor for the this isolation of bacteria. It required minimum 6 to 8 weeks, so the diagnosis is very difficult. When we talk about the status of John's disease in the world, so the disease is more common in the Europe and Western countries, in US and in most of the European countries. But when we talk about India, it has been reported that the disease is rare with the rare reports. So these are some figures actually. The table is showing some figures for the global herd prevalence. We can see here about the 60% of the herd from New Zealand are infected, and Netherlands have 31 to 71% herds are infected. And in the US, we can see 2.6 to 72% herds are infected due to this disease. And when we talk about India, the disease has been reported in the different parts of the country, and it is ranging from 10 to 5 10 to 69% in goats, 7.9 to 46% in sheep, and cattle 4 to 37%, in buffalo 3.9 to 30%. Very recently, Dr. Surveer Singh from ICAR, Indian Council of Indian Council of Agriculture Research, from the Central Institute for Research on Goats, have published their data of 20 years study, and they reported that about 23.3% of animals are infected due to this disease in our country, and they have reported that the disease is available in all the zones of our country. So when we talk about the public health significance or the risk to the humans, so the humans can acquire the infection from any infected animals through their food chain by consuming the milk, contaminated milk, meat products, and environment and water supply. So we can see here that the map is able to survive in the environment up to 246 days, depending on the specific condition, and it is also able to survive in the pasteurization temperature. And the presence of map has already reported in the protozoa, so it is very much have the some good survival tactics to survive in the environment. And when we talk these are some figures actually which has been reported the presence of Mycobacterium paratuberculosis in raw and pasteurized milk in different countries. We can see here these are some the statistics. It is there that it has been reported in many countries in UK, USA, Czech and Republic and others. So when we talk about India, the first reports on the presence of Mycobacterium paratuberculosis in the unpasteurized or pasteurized milk has been come out from the laboratory of Dr. Singh. So we can see here that the contamination of Mycobacterium paratuberculosis was there in the commercial pasteurized milk and unpasteurized milk, and even in the milk products, just like the flavored milk and ice cream. So very recently, this paper has been published in 2018 in Journal of Experimental Biology and Agriculture Science. That the most of the Indian brands, which has been very frequently available in the our local market, has some positivity rate is there for the presence of Mycobacterium paratuberculosis. So So these are some serological survey which has been conducted in the different time period in our country using the some indigenous ELISA kits, and they have found that the about 23% of the human population from different parts of different region of our country are infected. They are exposing have the antibodies. Actually, we can say that they got the disease or they have the disease because this population is exposed. We can say that this population is exposed to Mycobacterium tuberculosis, paratuberculosis. Some molecular epidemiology studies have indicated that the predominance of Mycobacterium bison type is mainly circulating in the human and animal population. So we can say that the human is acquiring the infection from animals. And if anybody asks me that is there any need to worry if we are getting the exposure of Mycobacterium paratuberculosis, then I would like to say yes, because the pathogen is mainly associated with a number of human diseases. We can see here this is the list of diseases which has been associated with the Mycobacterium paratuberculosis in human. So in present discussion, I would like to focus only the Crohn's disease, and this is the inflammatory conditions of the intestine, and this is a very we can see here that the lesions are occurrent at any location of the gastrointestinal tract of our human, and the active disease is mainly characterized by the reduced appetite, abdominal pain, weight loss, bloody diarrhea, vomiting, and tiredness. And we can see here this is the picture of Crohn's disease. So we can see here the corrugations and thickness of the intestine, and this is the ulcerative colitis. So when the disease at the early stage, so it is very difficult to differentiate the Crohn's disease and ulcerative colitis. So about the status of Crohn's disease in the world, the disease is also was very prevalent in Western countries, but in India, it has a lack of information. So when we think about the colitis and Crohn's disease in India, the first report on the incidence and prevalence of ulcerative colitis in Punjab was reported by Dr. Sood in 2003. And he reported that about 44.3% population were having the colitis in Punjab. So, however, the information on Crohn's disease was not available on that time with this study. So, very recently, I have gone through the surgical survey of Crohn's disease in India. We found out that the number of cases is increasing day by day in our country, and we can say here that some cases of the Crohn's disease may be misdiagnosed as a mistherapy as the intestinal tuberculosis in our country. So when we talk about the etiology, so there are a lot of hypotheses is there about the genetic predisposition and autoimmune theory is there, and the battle when the cause of the Microbacterium as the paratuberculosis as the Crohn's disease is about 100 years. There is some dispute in this etiology, and even this is not proven that. However, we can say here why the MAP is a strong candidate for the Crohn's disease. The first indication is come out from the this histopathological similarities. We can see here that the pathological similarities are very similar between the John's disease and Crohn's disease, and in situ hybridization is method has already reported to be presence of Microbacterium paratuberculosis in tissues of Crohn's disease patients. Detection of MAP in biopsies of Crohn's disease patients by culture, PCR, and probe hybridization has been proven, and immunological and therapeutical aspects has already proven the presence of paratuberculosis as the cause of Crohn's disease. And the partially fulfillment of postulates has already been proven. What the disease has not find out the etiology still. The presence of MAP in the inflammatory bowel disease has been reported in various countries. We can see here that this is the ulcerative colitis and this is the Crohn's disease. So we can see here that the significant higher number of patients have the Mycobacterium paratuberculosis in their intestine as compared to the ulcerative colitis. But the critics claim that some studies have failed to find out the failed to report the presence of Mycobacterium paratuberculosis intestine, and the critics explain that this is might be due to the spiroplast-like forms, which has been hard to isolate. So that's why these studies were failed to isolate the Mycobacterium paratuberculosis from intestinal tissues of the Crohn's disease patients. These two studies has been very supportive as the causative agents of Crohn's disease of Mycobacterium paratuberculosis. These studies were published in Lancet by Dr. Nasir, who reported the presence of Mycobacterium paratuberculosis in blood and, and in the breast milk of Crohn's disease patients. So these studies mainly indicated that the infection of Mycobacterium paratuberculosis in Crohn's disease patients is a systematic infection, just like the paratuberculosis infection in animals. So these studies has contributed to the significantly contributed to the evidence that MAB might be a cause of Crohn's disease in human beings. So this is the first study which was come out in 2008 from our country, that the Crohn's disease patients and UC patients was screened for the presence of Mycobacterium paratuberculosis using culture method, and reported that 80% of cases were having the Mycobacterium paratuberculosis in Crohn's disease patients. And however, the 12.5% cases were also healthy humans were also having this bacteria. So this is some caused some dilemma that the disease is caused by the Mycobacterium paratuberculosis. And when we think about the genetic aspects of this bacteria, so we go for the detection of Mycobacterium paratuberculosis in animal attendants, which are very close to the infected patients, and we found out that the duration of the association is associated with the presence of Mycobacterium paratuberculosis in the stool of the animal attendants. So this again already indicated that the bacteria can be associated with the causation of Crohn's disease in human beings. However, after 1 year, another study has been published in Indian Journal of Gastroenterology. They by Madam Sasikala, and they screened about 186 DNA and 24 RNA samples from Crohn's disease patients, and they don't find any evidence of Microbacterium paratuberculosis in Crohn's disease patients. So this study already not supported the hypothesis that Crohn's disease is associated with the Microbacterium paratuberculosis. So more studies are required in this area to clarify the etiology of Microbacterium paratuberculosis. I would particularly highlight one study that has been conducted by Dr. Ichi Mumtani in Japan. Actually, this study is mainly based on the impact of evaluate to how the microbacterial antigens, lipophilic antigens, are associated or have the potency to develop the Crohn's disease in mice. So they have used this TNBS model. This is a very common model to for the experimental analysis of Crohn's disease. So this is the normal colon, and we can see here this is a TNBS induced mucosal ulcers. So when they make a study of the they take the experimental animal mice in 10 weeks old mice, and they have the lipophilic antigens at the different antigen concentrations, and they have two controls, single dose control and non-antigen controls. So they give the antigen and controls by the transanal routes, and after 5 days they collect the colon tissues and go for the histological examinations, and they found out that the group 1 and group 1 to group 4, these were the experimental groups actually, they have developed the Crohn's disease like pathology, and the single dose control and non-antigen controls were not having any types of histological differences. So the studies mainly provide the evidence is that the MAP antigen has the potency to induce colitis. That is very similar to mouse TNBS induced colitis, which has been used as a experimental Crohn's disease model. So when we talk about the treatment, so there is currently no cure for Crohn's disease, and there is no one treatment that works for everyone. The goal of medical treatment is to reduce the inflammation that triggers your sign and symptoms. Treatment for Crohn's disease depends on how severe the symptoms are and how much of the gut is affected. During surgery, your surgeon remove the damaged portion of the digestive tract and then reconnect the healthy sections. This is also a fact that nearly 80% of patients with Crohn's disease require surgery, again, this surgery in their lifetime. So when we talk about the COVID-19 and Crohn's disease, because we have listened a lot about the COVID-19, so I'm not going to highlight the background of this COVID-19, and but I would like to mention that the two disease conditions are unrelated actually. But the pupils with Crohn's disease should be aware of certain factors regarding COVID-19. A person's general health may play a role in the risk of COVID-19. Additionally, some treatment options such as immunosuppressive drugs may increase the risk of infectious illness such as COVID-19. Currently, pupils with IBD who do not have severe inflammation in the intestine do not take any immunosuppressive medicines, and they are not malnourished and should have about the same they have the same level of risk of COVID-19 infection as the general population. And the people who have take the immunosuppressive medicines may be at the increased risk of infectious of the COVID-19 and maybe the serious complications of the infections. However, pupils taking these medicines should not stop taking them unless it is under the direct guidance of a doctor of their so for the management of the inflammatory bowel disease, which includes both the ulcerative colitis and the Crohn's disease during the COVID-19 pandemic, has been issued by the American Gastroenterology Association, and they especially mentioned that the patients with IBD should continue IBD therapies and infusions, and having IBD does not appear to increase the risk of SARS-CoV-19 to infections or development of COVID-19. And the instructions has been given to the patients who have already exposed or have the fever, respiratory symptoms, and digestive symptoms. They have to stop the thiopurine in their treatment and stop biological therapies, and they can also restart the therapies after completion of the resolution of the COVID-19 symptoms. So these things needs to be taken into considerations for the IBD patients. And, thank you very much. Jai Hind.
Thank you. Thank you very much, Dr. Ajay Singh. It was a most informative and very well organized presentation. I must compliment you for this beautiful, very excellent, informative presentation. In fact, personally, I must admit, I I learned a lot. Really,
Thank
your
you
presentation was
for
so
your
so informative, and you brought it at the end also the its relevance in today's time when we are only talking about COVID-19. Of course, I mean we need to be uh, all prong approach has to be taken, but at the same time, like you have mentioned, the risk of other diseases which we at times tend to overlook. For example, most important myobacterium diseases in humans, as you have mentioned rightly, leprosy and TB, tuberculosis. We tend to we tend to believe that these are now almost, at least leprosy, people think that it is eradicated, kind of eradicated. You have shown that the rate has really gone down, and maybe the rate is less than 1%, but 10,000, but yet we are still to reach the level that we can say that we have been able to eradicate it completely. Also, you have brought forth the importance of MAP diseases in the animals, particularly in the ruminants, and you have very eloquently made the presentation about uh, the point which I feel which emerges from your presentation. Very important point is that in many important areas there is lack of information, like you showed for different countries where we have a very systematically done studies were made, and we have that kind of data which is lacking in our country. You have shown some data which have been done by people like in certain parts of northern India, certain places, some place in Rajasthan, some place in Punjab. But if we have to have a national database, perhaps that is still lacking. And I think that is one area. Simultaneously, of course, the etiology, you have also mentioned about it. But the starting point should be a national level data to be available, and accordingly a strategy to be derived so that we can focus on the priorities are identified, and we can focus on those areas which need immediate attention, which are equally important for the human population as much as the animal populations, because these are interlinked, particularly in a country like India, where our population depend hugely for their protein intake. They depend hugely on the milk proteins. And some of the data, some of the tables that you have shown, even the chances of transmissions or chances of causing inflammatory bowel syndromes or bowel diseases are pretty high, and people should be made aware, and necessary steps should be taken. And I think once people are aware, they will take the necessary steps so that it can be contained in a significant manner to a large extent. And this kind of study that you are doing, Dr. Ajivir Singh, I once again compliment you hugely. Very well presented work, and we have gained a lot from your presentation. Thank you very much.
Okay,
Thank
thank
you.
you, madam. Thank you.
So, Anshuman, do we have our second speaker? Would you kindly
Yes, ma'am. Yes, ma'am.
introduce?
There are a few questions from people. I'll just showcase them and
Yeah,
just we'll have a
if there are
reply
questions,
from.
I didn't know. Yeah,
Yes, ma'am.
yeah. I didn't know about the
Just
questions, that whether we can take questions or not, but I understand that there is a time frame for that. So please go ahead. Those who have any questions.
okay. Yes, ma'am. I don't think so. There are any questions like I have asked people, but nobody is replying. Okay,
Yeah, I
one
think his
question is coming.
presentation was, yeah.
Completely. Yes, its presentation was complete. One question is coming that what is the present scenario of COVID-19?
Okay.
What?
So, Dr. Ajivir Singh.
Presently, we can say that the present scenario is under control, and as we know, we all of know from the media and other there that cases are increasing. So we have to take precautions, and we have to be stay at home as much as possible. So, but the situation is under control now because my laboratory is mainly involved in the diagnosis. So in last few days, we have getting a number of samples for testing, but the positivity rate has been declined. So this is a good signal actually. So we would like to thank that the people are staying home, and if you have any urgent work or any necessary work, then you have to go outside. Otherwise, please try to be stay at home as much as possible.
Okay, thank you very much, sir. It was a very informative session, and I guess everybody has learned a lot from this session. Thank you very much, Dr. Ajay Singh,
Sure,
sir.
sure.
Thank you, sir.
Thank
Thank
you
you.
very much.
Thank you, everybody.
So, do we have our
Yes.
second speaker, Dr. Ulrich Berg?
Yes, we have with
Anshuman?
us our next speaker, our lead speaker and guest, Dr. Ulrich Berg from Germany. And Dr. Berg is the president of German Association of Homotherapy, Germany. And today, Dr. Berg will be giving his lecture on how can Agnihotra help with COVID-19 crisis. Dr. Burke has also received several awards worldwide in his studies and in his presentations. Dr. Burke will just work to give the lead lecture on how can Agnihotra help with COVID-19 crisis. Handing over the mic to Dr. Burke.
Welcome, Dr. Ulrich Berg. Welcome to this webinar session. And we are eagerly waiting to listen to your presentations and learn how we can fight the problems or how we can combat COVID-19 using Agnihotra or homotherapy, which you have been practicing and studying at different levels for many years that we know. So we welcome you and we invite you to make your presentation. Dr. Ulrich Berg.
Okay, thanks everybody for inviting me to this webinar. And
Yes, sir. Your slide is visible. Your slide is visible.
the slide is visible. Okay, that's very
Yes,
good.
sir.
Yeah.
And also it changes. That was the problem last time.
Yes.
Okay, so
Okay.
let us begin. Again, good morning, or for you, good afternoon. Here it's still quite early in the morning. I'm in Germany. Good afternoon, everybody. So the
Good
subject
afternoon, sir.
is how can Agnihotra help with COVID-19 crisis? And now you all know, and just now the last speaker said best is to basically stay home. Everybody is now affected by this COVID-19 caused by the coronavirus SARS-CoV-2. And still many states, countries are in a lockdown. And even after that will be over, we have to be very careful. So what can we do to protect ourselves and our families, and how can Agnihotra help? It is very interesting to see that recent scientific studies have shown that COVID-19 is much more severe and deadly in areas with high air pollution. And second thing, this coronavirus can be transported by particles of air pollution. So if you then see that Agnihotra purifies the atmosphere, so if air pollution is a very important factor for severity of COVID-19, then Agnihotra can be a useful tool to combat the coronavirus. As this disease is pretty only occurred recently, we do not have yet scientific studies directly on the effect of Agnihotra, but there's a lot of preliminary evidence which makes it likely that Agnihotra can help. How could Agnihotra help? There are three ways. One is Agnihotra could help to reduce the probability of getting infected. Second, it can reduce pre-existing conditions, thus leading to a less severe cause of COVID-19. And third, Agnihotra can help to support the body's immune system, and the immune system is most important to overcome the infection. Let us start with one, reducing the probability of getting infected. There's one report from Spain, from Madrid. At that time, Madrid was the hotspot of COVID-19 in Spain, and there were many, many people infected and thousands of people died. There was one person, Elizabeth, and her partner. She lived in a house together with a third person. And this third person, he runs a restaurant in Madrid, very popular restaurant. So he had contact to many, many people. And when he got tested positive for coronavirus. Then Elizabeth and her partner, they were concerned, and they got tested also, but nothing happened to them, and the test was negative. And the doctor who did the test said that's practically impossible because they were living together in the same household, shared the same kitchen, shared the same bathroom, had a party together just a couple of days earlier. So they said nearly impossible. So what could have helped her? The only thing which makes a difference is she and her partner, they both performed Agnihotra regularly. So it seems that this has helped them to avoid getting affected. Let us look at the ways how the virus is transmitted. There are three main routes. One is touching surfaces like door handles and so on, which an infected person has touched before. The virus remains on such surfaces for a couple of hours, on metal maybe up to 10 hours. But still, that is not the most likely way. Reports in Germany, some studies in Germany had showed that the virus may be there on door handles and so on, but it's not in an active form. It cannot be revitalized. So maybe that is not the most dangerous way. More dangerous seems to be the transmission through air. There are two ways. One is respiratory droplets created by infected people who cough or sneeze. And these droplets stay in the air for a couple of minutes. And if you then either inhale them or they come to your eyes, then you can get infected also. So that is one way, but the second way was found recently by Japanese scientists, and they said that there are mini droplets. They had cameras which are with very high resolution and very fast, and they could show that there are droplets which are caused by people when they speak. So they don't have to cough, they don't have to sneeze. Just by speaking, these mini droplets are in the air, and they also carry the coronavirus. So now let's talk about Agnihotra. We have seen that in many studies that Agnihotra purifies the air. For example, the number of bacteria in the air is reduced considerably by just performing Agnihotra in a room. So, very simple experiment. You take a petri dish with some nutrient solution, open it for 10 minutes, close it. and then you send for incubation. And then you perform Agnihotra in the same room, and after Agnihotra, you open a second petri dish, open for 10 minutes, send for incubation, and then you compare, and this is what you see here. You see a very big difference between before Agnihotra and after Agnihotra, the number of bacteria. Now, this holds for bacteria, but as both bacteria and viruses travel on particles of air, the same is likely to hold true for viruses also. And here you see the statistics show, which shows a big reduction in microbial population in the air before Agnihotra, after Agnihotra. They got a reduction of, say, 70%, but that is only by one Agnihotra. And if you perform Agnihotra in your household regularly, then it goes down more. We are doing more studies when the lockdown situation allows that. So that was about microbial load in the atmosphere. Now about the other pollutions of the air, chemical pollution and physical pollution. Chemical pollution is SOx and NOx mainly. Physical pollution is mainly the particulate matter. And you see here that after Agnihotra, these values go up a little bit, but then they come down to say 30 or 40% less. And that's again just one Agnihotra. And so some people said, okay, normally anyway, during the night, because we did that in the evening, particulate matter will be less because particles will settle down. So we did the same thing from morning to evening, and we got the same results, same reduction of 30 to 40%. And again, that was just one Agnihotra, and we want to do a replication of that experiment and just plan it for 1 week. Now, what happens? There was one explanation given by an American physicist about the reduction of particulate matter, and he said that the colloidal molecules of ghee and cow manure produced by Agnihotra, they can gelatinely attract and grab pollutants in the air, similar to the way how water is purified by being flocculated. And he says physically, because of the ghee and the manure, the smoke would be electrically charged. I come to that a little later. And as I already mentioned, as both bacteria and viruses, so to say, travel on aerosols, a reduction of these aerosols automatically removes bacteria and viruses from the atmosphere. I just mentioned, because this physicist said that because of the burning of cow's ghee and manure, the smoke would be electrically charged. This experiment we did in Germany long time back, and it was, and we know that negative ions in the indicate fresh air. So if there are less negative ions, that's an indication for polluted air. And normally, every smoke reduces the amount of negative ions. So when I went to this engineer to do the test, and I told him about Agnihotra, and he asked, "Oh, will there be smoke?" And I said, "Yeah, there will be some smoke." And then he said, "No, we don't have to do the experiment. Any smoke will reduce the count of negative ions in the air." So I said, "Okay, let's try." And then in order to prove his statement, he just took his cigarette and blew the smoke of the cigarettes towards the instrument, and yes, the indicator went down. And then when we performed Agnihotra, the indicator went up. So negative ions were more. He was very much surprised. And that now can be very interesting also, because these negative ions in the air, they can have an effect on microbes in the air. And This is one thing which we studied in some college in Pune about the virulence of bacteria. And the result was the virulence of bacteria was going down if they were in contact with Agnihotra ash. The capsule formation, hemolytic activity, and pigment formation, all these properties which are relevant for the virulence of bacteria, they came down. And here you see the pictures. That's the capsule formation, hemolysin production, pigment production, all went down. And that leads to a reduced virulence of bacteria. I come to the effect on viruses in a minute. Just one more thing. Effects of Agnihotra ash on multi-drug resistant bacteria. You know that now in the last decades, multi-drug resistant bacteria, that's a big problem in all hospitals worldwide. And if they have these multi-drug resistant bacteria in the hospital, it's very difficult because they don't have any standard way of treating the patient. And often patients come to a hospital, maybe they have broken a leg, they come to the hospital, they catch these multi-drug resistant bacteria, and then they uh, and then they get an infection, maybe pneumonia, and then they often die not from the disease for which they came to the hospital, but for the disease which they catch in the hospital. So that's very important if we can deal with the multi-drug resistant bacteria. So what they did, that was an experiment done at Kathmandu University, Nepal, and they had a multi-drug resistant E. coli, and they had one total control, just these E. coli, one with Agnihotra ash, one with control ash. And you see here the difference, Agnihotra ash and control ash. Agnihotra ash, much less of these bacteria. And here you see the numbers. After 5 days with the treatment with Agnihotra ash, from the count of 2,400, the count came down to less than 3. After 3 days, it came down from 2,400 to 220, means 10%, less than 10%. Now, there are many studies of effects of Agnihotra and Agnihotra ash on bacteria, much less so on viruses. And one reason for that may be that normally bacteria, which we are studying, they can be cultured in artificial culture media and then observed for research. Yes, we know not all bacteria can be cultured. So about these bacteria also, we just know less because they cannot be cultured. So it's much more difficult to do some research on them. And also for viruses, that is different because viruses, they are parasitic and they need living cells to multiply. So putting them on some nutrient solution wouldn't help. They would not multiply. So they need a living host body for growth and multiplication. But one thing is, and that is very interesting, we could show that there's a reduced virulence of bacteria when in contact with Agnihotra ash. So about viruses, see, the coronavirus has some integral membrane protein which is involved in several aspects of the virus's life, such as assembly, budding, envelope formation, and so on. And if this membrane is damaged, then the virus material cannot sustain. Can Agnihotra damage the protective protein layer of the viruses? Let us go back and remember this study which showed the loss of capsule formation of the pathogenic bacteria Klebsiella pneumonia when exposed to Agnihotra ash. So, the ability of the smoke of Agnihotra fire to destroy the cell wall of bacteria uh, must be also applied to the protein membrane of protection of the viruses. So that is a strong indication that this virus also could be uh, destroyed by Agnihotra and Agnihotra ash. Okay, that was point number one. Number two, reducing pre-existing conditions, thus leading to a less severe cause of COVID-19. Uh, so what is if people already have contracted the coronavirus? We have seen, and there are many studies, that the disease is more serious in certain high-risk groups, that is people who have any of the following pre-existing conditions, either heart disease, high blood pressure, asthma, emphysema, chronic bronchitis, diabetes, cancer, immune system deficiency. And most people who have died from COVID-19 were suffering from one or more of these pre-existing conditions. Agnihotra can help to reduce these pre-existing conditions. Actually, it is normally they say mainly older people are affected by the disease, but that is because older people normally they have one of these pre-existing conditions, so they are more prone to that to a severe cause of COVID-19. What can Agnihotra do? A few examples. Agnihotra regulates the blood pressure. We have seen people with high blood pressure, they have a higher risk of severe cause of COVID-19. Here, that was a simple experiment done some time ago. We had a group of 10 people. Some of them had high blood pressure. For them, blood pressure came down. Some also at the bottom of the table had low blood pressure and blood pressure came up. So Agnihotra regulates the blood pressure. And if you have a normal blood pressure, then your body will be stronger to fight the COVID-19 if you have contracted. Some other examples, heart disease. You know, this lady, she was very ill, and her electrocardiogram showed that part of the heart was dead and normally cannot recover. But then she was performing Agnihotra, and after 4 years, she took an electrocardiogram again, and the heart was totally fine, and no sign of a pre-stroke, no sign of part of the heart dead. So that was very astonishing. Then another of these pre-existing conditions is asthma. Asthma is very important because COVID-19, first it attacks the respiratory system. So if you have asthma and your lungs are already affected, then you have more problems to deal with this disease. So this lady from US, she was very sick, and she went to a lung doctor, and the lung doctor said that the x-ray showed that part of the lungs were completely black and only a small part was left over, which still worked fine. When she performed Agnihotra, uh, after a short time she stopped taking the medicines, and after 3 months, she went to the doctor, and the doctor took an X-ray of the lungs, and he said, "Okay, I don't understand. I don't know what you did, but your lungs are totally clear, and no more medication necessary after that." And till now, and that's more than 30 years ago, when that happened, till now she is fine, no more asthma. That is another case of asthma. This lady, she was also suffering from asthma, and she she often had to go to the hospital in the middle of the night to get some shots in order to be able to breathe. And when she started performing Agnihotra, then the asthma went away. Within 2 weeks, it was cured. And since then, even 40 years later, she did not have get it back. Diabetes, that was a farmer. He performed Agnihotra just for his farm. And we know this chemical farming has the side effect that farmers get sick. Just think of the cancer train from Punjab. Here, there's a positive side effect of Agnihotra. This guy had a blood sugar level of more than 300, and it came down to 200, and later on even to 100, just by him performing the fires. Prostate cancer got healed, lung tumor got healed, and we have so many cases where the immune system also got stronger. And immune system is most important for fighting a virus infection. One more very important point is stress and anxiety. If you have stress and anxiety, then for all kinds of diseases, that is aggravating. That makes it more difficult for the body to get healed. So how can we reduce our stress? And according to the Vedic knowledge, Agnihotra attracts, balances, and harmonizes prana. And prana and mind are thought to be like the two sides of the same coin. So if there are beneficial changes in prana, then automatically there are beneficial changes in the mind also. And there are several studies which show that Agnihotra, if performed regularly, it reduces stress, reduces fear, depression, and anxiety. And that leads to a very strong boost in the immune system. One such study was done in Moscow in Russia. And a group of people came for Agnihotra for 2 weeks regularly. And you see the change in anxiety and depression. The red bars show anxiety before Agnihotra. The white bars show anxiety after. You see quite a considerable reduction between 40 and 60%. And then again, the black bars show depression before Agnihotra, the green bars depression after. You see again a very considerable reduction. Another experiment was done, that was a control experiment now in Medical University in Santiago de Chile, South America. And these students of medicine, they were just before a very important and very difficult exam. So all of them were very much stressed. So the experiment took 100 of these students and they measured their stress level. And the stress level was very high. And then they took the 24 of these students with highest stress level and divided into two groups. And both groups were invited for some Tai Chi lesson. And they were told, okay, now we do some Tai Chi and that reduces the stress. And what these students did not know is one of the groups, when they did Tai Chi, Agnihotra was performed in the next room. So they did not know about that. They did not see Agnihotra. They just thought they were doing Tai Chi. And then the stress level was measured, and you see 100% of the students before this intervention showed extremely or severely stressed. And that came down to 41.7% for the group with Tai Chi. So Tai Chi helped to reduce the stress. But the group where Tai Chi was done and Agnihotra next room, only 8.4% of them had still severe levels of stress. So it came down much, much more compared to the group with only Tai Chi. And that is a double-blind study, so that shows a very good result. Of course, the result would have been much better if the students could also have performed Agnihotra. And we are thinking of how to do it in such a way that the placebo effect can be excluded. It's a little difficult, but it's possible. And here the same study, the first study, they tested the students, the stress level with some questionnaire, and here they tested the students by examining the cortisol level in saliva, and you see the same thing with Agnihotra. It came down much more. This is another study, and that was done again in Moscow in Russia. And what they did is they did the EEG before Agnihotra and after Agnihotra. Before Agnihotra and after Agnihotra. Now look at the green area here. This is the beta waves. And the beta brain waves, they indicate some activity, maybe also stress. And the alpha waves indicate calm, some calm activity, calm state of the mind. Now here, the beta wave is 70%, and after Agnihotra, it comes down to 22%. And the alpha before Agnihotra was 17%, and after Agnihotra, it's 58%. Means it's increased a lot, showing that Agnihotra really reduces stress. So we have seen several such studies. Now one more, Kirlian photography. Kirlian photography, you may have heard that. You put your fingers on some special screen, and very high voltage is applied, and then you see some kind of like energy lines coming out that shows the aura body of the person. And here before it is before you see, then the lines are very much interrupted, and then just 3 minutes after Agnihotra, just one Agnihotra, you see the aura field around this finger is much more homogeneous. And that was the person with depression. You see the aura is very dark. And after just one Agnihotra, you know, it already is much more complete. Left there big holes, and at the right side, after Agnihotra, it's already much healed just by one Agnihotra. This again shows that the mental state of a person is much improved by Agnihotra, and this mental state of a person is very much responsible for the immune system. Now, we have two studies on viruses itself. Okay, this study, it's not on the coronavirus. As I said, coronavirus is a pretty new thing, so we have not finished a study yet. But it was on HIV/AIDS, and this was a group of children in Mysore, Karnataka, and they were in a very bad health state when I came there, and the doctor said that they were not able to walk much, and they had to be driven by a bus a few hundred meters to school, and often in the night he had to come for some infection. To treat these people and so on. Then they started to perform Agnihotra. All the children themselves performed Agnihotra, means each child had his or her own pyramid, and they were performing Agnihotra morning and evening. And then after some time, you see the viral load went down a lot, and also the CD4 count improved, and some other ratios also. And also their general health was very much improved. Now, direct effect of Agnihotra on viruses. I mentioned before, with viruses, studies are more difficult than with bacteria, because viruses do not replicate outside other cells. They need other living cells for that. So you can't just add Agnihotra ash to the viruses and see what happens, because nothing is happening anyway. So there was one lab test with some other kind of viruses, also coronaviruses, but not these novel coronaviruses responsible for COVID-19, because that was done last year. We didn't have this corona COVID-19 virus yet. So Agnihotra, and you can't add in cell lines, you cannot add Agnihotra ash directly. Whatever you add, it has to be filtered. So what they did is they took Agnihotra ash water, means kept Agnihotra ash in water, waited for 24 hours, and then filtered that. So the ash was no longer in the water, just the water which was in contact with ash for 24 hours was used. And that was injected into the cell line, and they found a reduction of virus production of 50%. Now, 50% may seem not to be very much, but still, that could make the difference between a mild and a severe progression of the illness, or between no disease at all and a mild progression. Now, we can expect that with other types of coronaviruses, like these novel coronaviruses, we could see similar results, as we also have seen with the HIV virus. But for that, some more research is necessary. One last example, which just shows the power of Agnihotra. Remember this Bhopal catastrophe in 1984, when MIC gas leaked at the Union Carbide factory in Bhopal, and thousands of people died, and tens of thousands of people were heavily injured. But those people who lived close to the factory, and there were several families, they performed Agnihotra regularly, and nothing happened to them. So it seems that Agnihotra proved to be to produce a very protective armor for all of them. Now, what is Agnihotra? I want to make clear when I talk about Agnihotra that we understand the same thing. It's the smallest and basic Homa healing fire, tuned to the specific biorhythm of sunrise and sunset. And here you see the utensils. You need a copper pyramid of fixed size and shape. You can't get it on the market. On the market, you find the other types. It has to be exactly that one. What you burn is dried cow dung, pure cow's ghee, some unbroken, unpolished rice, akshat, Sanskrit mantras of a certain vibration, and a timetable which gives you exact times of Agnihotra, sunrise and sunset. What you do is you light the fire, and at the time of sunrise sunset, you already should have a flame which is fully ablaze. And you take a few grains of unbroken rice, apply a few drops of ghee, and then you make two portions, and then there are two mantras, and after swaha in these two mantras, you add one portion each to the fire. So the mantras in the morning, Suryaya swaha, Suryaya idam namama, Prajapataye swaha, Prajapataye idam namama. And in the evening, Agnaye swaha, Agnaye idam namama, Prajapataye swaha, Prajapataye idam namama. That's it. Only takes 5 or 10 minutes in the morning, 5 or 10 minutes in the evening. Why these mantras? Why exact time? There is some description giving in ancient knowledge about sunrise, and it says that at sunrise, many fires, electricities, ethers, and more subtle energies come all the way from the sun to the earth. And this flood of energies has purifies everything, and it has a subtle music in it. And the quintessential sound of that music is the mantra which we utter at that time. So that explains why exact time, because this flood of energies is there only for a very short time span. And second, why the mantra, because that leads to some resonance effect. And what happens, according to ancient knowledge, a channel is created through all the atmosphere, and prana energy, which lies above the atmosphere, comes down through this channel. It creates an aura energy field around all plants. And when the flame dies, this energy comes back and is kept in the Agnihotra ash. This is why the Agnihotra ash has healing properties, can be used in medicine, can be used for plants, for animals. Okay, now as the problem of COVID-19 is still there and not so much research done so far, we suggest some scientific research for things. One is if we have a group of COVID-19 patients, then best would be if these patients would perform Agnihotra themselves. And if they are not able to do that, somebody does it for them. And then we compare with other patients where there's no Agnihotra. Then second, we measure the concentration of coronavirus in the air in some hospital room where you have several corona patients. And then you perform Agnihotra and measure again. Also, you could check the virus concentration on surfaces in the hospital before and after Agnihotra. Then, what I mentioned before, testing the effect of both Agnihotra and Agnihotra ash on the coronavirus in cell lines. Before that was some other type of coronavirus, but it should also be done with this novel coronavirus. And not only with Agnihotra ash, because as I said, it is difficult. You cannot apply the ash directly to the cell lines because it has to be filtered. So some part of the effect is not there. So it would be also good when you have the cell lines to actually perform Agnihotra there and see which effect that has. And then one very important point, because doctors and nurses who work in such hospitals with COVID-19 patients, they are much affected, and worldwide many of them get seriously ill, and many also have already died. Many means thousands and thousands worldwide. So it would be also very good to do some trial uh, aimed especially at this group of medical professionals to see how Agnihotra can help these people who risk their lives in order to help others. Here are some references. That is my contact. You can see the email address, especially on the web page. And that's it. Thank you.
Thank you very much, Dr. Ulrich Berg, for a very interesting talk, a very interesting presentation. So before I conclude this session, we would like to invite if there are any questions to the speaker. I think there is already one
Yes.
question. So would you like to
Dr.
first
Berg,
answer?
there are a lot many questions for you and comments as well. A comment from Alok Kumar is coming. Dr. Berg, you are amazing. Uh, okay, so here a question for you is, what are the disadvantages of long-term anxiety? I repeat, what are the disadvantages of long-term anxiety?
Anshuman, may I intervene here? I think
Yes, ma'am.
because we have to finish in time, there is another lecture, valedictory session. So please take the questions which are directly related to this talk. Like he's asking about the
Sure.
long-term anxiety, disadvantages of long-term anxiety. That is something not directly related. If there are any questions which are related to Agnihotra or homotherapy per se, I think Dr. Ulrich will very much agree, I'm sure, that that will be more pertinent. So, if there are any such questions.
Okay, can I just
Yes,
say
ma'am.
one sentence to that?
Yeah,
Uh, you're
Yes,
right.
sir.
I don't
Yes.
want to give a lecture on disadvantages of long-term anxiety, but just one suggestion. If you have that problem, start performing Agnihotra regularly. The problem will go away.
Okay. The
sure.
next question is from Dr. Poonam Mehrotra. There's a mixture of neem leaves and flour, camphor, ghee, dry cow dung, cloves, dry rose petals, green cardamom, black till, and Havan Samagri works as Agnihotra.
See, at the end, I explained when I talk about Agnihotra what the procedure is. It's a very simple procedure, and it does not have this Havan Samagri and so on. It's just cow dung, ghee, and some grains of rice. But most important, exact vessel, exact size and shape, exact timing, sunrise, sunset, exact to the second, and two mantras only, morning, two mantras evening. That's it. And we, with that Agnihotra, we have seen a lot of all these effects, the beneficial effects. If you do some other kind of fires, the effects, I don't know. You can try yourself. But try that and you will see very good results.
One more question is coming from Pradeep Garg. He's asking, sir, what is it working biodynamic as Agnihotra as?
I don't understand the sense. It's dramatically something. I mean, biodynamic, I know that. I know biodynamic in farming, and I can only say biodynamic is great, but at the time when it was developed by Rudolf Steiner 100 years ago, we didn't have this problem of air pollution. So if you are biodynamic farmer, great, just add Agnihotra to your procedure. That will take care of the new problem of air pollution, which affects all plants also. So it's a good combination.
Okay.
Anshuman, if there are no more questions, I have one
Yes,
question
ma'am.
also.
Yes,
Dr.
ma'am.
Ulrich. Dr. Ulrich, your presentation, as I said, was very fascinating. It reminded me of one of our senior former colleagues, Dr. R.K. Pathak, who did a lot of work on Agnihotra and Homa on agriculture, particularly. I was just, you gave a very good, what should I say, kind of a logical explanation how sorry, how Agnihotra purifies the air. My question to you is that since you have been studying this Agnihotra and the homotherapy, your association and you yourself for many, many years. So, what is the size of the Homa, and what is the duration of Homa, which will purify, say, the air in, say, 1,000 cubic meter? Because when we are talking about its chelating effect, it will be a certain volume, I mean, certain particulates per given volume. So, for a volume of, say, 1,000 cubic meters, what will be the size of the Homa which needs to be performed to purify the air, particularly about the particulate materials?
Okay, first, nice that you mentioned Dr. R.K. Patak. He's a good friend, and we work together since very, very long time. So, about pollution, see, when I gave this explanation of how air pollution can be purified, uh, that is only, I would say, that is only one aspect of how air pollution can be reduced by Agnihotra. And for that, yes, we need the smoke for that, gelatingly crabbing the particles and so on. Just I want to mention one experiment we did on purification of water, because it's interesting because there's no chemistry involved. We took water in closed bottles next to Agnihotra, and they were purified. Means we measured especially the coliform bacteria, and they went down by, say, 70% in a couple of days. And so there was no chemical interaction with either ash or the fumes of Agnihotra. If you ask about the size of the Homa, the size of this vessel, the Agnihotra pyramid, is fixed. It is only, say, this size, not very big. And if you want to purify more area, you can't make the vessel larger. It is like with a music instrument, because Agnihotra is all about sound, vibration, energy, resonance. So if you would change the size, it wouldn't work. Similar if you give a concert with a sitar and the audience is very big, you can't uh, make the sitar bigger. Then, the music wouldn't be nice anymore. So same here. But especially for agriculture, we have a system of resonance pyramids where you have one pyramid in the middle where you perform Agnihotra, and then one in the northern, southern, eastern, and western side of the farm. And, you don't
Yeah.
perform Agnihotra there, but the energy of Agnihotra goes through the earth to this pyramid. It's a little bit more complicated, but just the main thing. So with that, the area which you can affect is up to 80 hectares, 200 acres. So that's quite a large area. And that has
Okay.
been shown in many instances, many farms in South America, Europe, and also in India.
Okay. So, if there are no more questions, it will be my pleasure to conclude this lecture also. We all compliment Dr. Ulrich very much
Thank
for
you.
this very fascinating presentation. And I mean, I am aware of the Agnihotra. As I said, I was quite I interacted many times with Dr. Pathak. And the things which are most shocking and most informative from today's lecture, I would say that the effect of Agnihotra on the control of multi-drug resistant bacteria, I think that is something worth pursuing more intensely, more scientifically, more at multi-location, multiple participants, multiple researchers, because that is something which is really causing great concerns for human health. And it is so common that people who go to the hospitals for something, maybe a cardiac problem, and then after, say, 3 days or 5 days stay in the hospitals, they come back with an infection and having some bacterial infection. Most common is E. coli infection. So I was very much impressed by seeing your some slides where you have shown that by performing Agnihotra, you can actually substantially reduce the populations of multi-drug resistant bacteria, particularly E. coli. Also, I was quite fascinated by your presentation that even when Agnihotra is performed in a adjacent room, there was significant improvement in the stress level and anxiety and depression of people who have taken the Tai Chi. Tai Chi itself is a therapy for reduction of such anxiety and depression levels, and the results that you showed in terms of the Agnihotra performed in the next room, that is very impressive. That I would like to relate or like to link to your last slide, where you suggested that this can be a strategy to provide some kind of better immunity, better power of immunity to the health workers, doctors, and nurses who are serving at the hospitals of COVID 19. So, in fact, they don't really have to do anything. It can be performed in a room, maybe a centrally located room, which around that, if the people who are working there, they also get benefited. I think that can be a very good recommendation from this session, which can be given to the concerned departments which can follow it. I mean, there is no harm trying. It is not going to harm in any way. If at all, it will be beneficial. And it's a very simple thing to do. The third point taken from your lecture is about, you talked about, I mean, that I think needs more detailed, deeper study, is about the impact of Agnihotra on the coat protein of novel coronavirus. I think that needs more studies and more in-depth studies, which may be again one of the suggestions to be taken up by the researchers who are in the field and studying and trying to find a solution to this pandemic. All in all, I think all of us are much benefited by your eloquent presentation, Dr. Ulrich, and we all thank you very much for this presentation. And with that, I think this session comes to a closure. We had two wonderful presentations by Dr. Ajay Singh and by Dr. Ulrich Berg, and both have given us much food for thought, and uh, we are much more enlightened and educated, both in terms of myobacterial diseases in the human and animal world, as well as the potential or possible use or benefits of Agnihotra or homotherapy in combating many of the diseases, many of the well-being problems, both for human and animal and the world in general, particularly in these critical times of Corona or COVID-19 pandemic. With that, I once again thank the organizers of the I.S.G.B.R.D, both the president, Dr. Rajendra Kumar, and the general secretary, Dr. Upadhyay, and of course, all other organizers, including Anshuman and all the speakers and all the participants. Thank you very much. It was my pleasure.
Thank you, ma'am, for sparing your valuable time, and you dissected the science at length, particularly Agnihotra. And I have also taken one work. I have to make research where Dr. Ulrich learned Sanskrit, because that's a kind of issue. Otherwise, foreigners usually don't spell out or pronounce properly as far as Sanskrit is concerned. So, Dr. Ulrich, I congratulate you. And ma'am, yeah, Anshuman, please go ahead.
Thank you, Dr. Malvika Dadlani, ma'am, for joining us in this session at this international webinar. I extend my heartiest thanks to you. Now, I
Thank
would like to invite our chief guest for the valedictory session and chairperson for the last technical session, Dr. Ranjana Agrawal, ma'am. Ma'am is the director of CSIR, National Institute of Science and Technology and Development Studies, NISTATS, since June 24, 2019, and also holding an additional charge of director, CSIR, NISCARE. Previously, Dr. Agrawal has served as Professor of Chemistry and Director, Women's Studies Research Center at Kurukshetra University, Kurukshetra. She is also actively collaborating with scientists from USA, Spain, and Ireland. Her research contributions have been acknowledged in the form of awards, notably Dr. Basudev Banerjee Memorial Award by Indian Chemical Society and Professor S.S. Katyar Endowment Award by Indian Science Congress. We warmly welcome you, ma'am.
Thank you very much, Anshuman, for the nice introduction. I would like to thank Professor Rajendra Kumar, who is chairperson of the organizing committee of this international webinar and also president of Indian Society and of Genetics, Biotechnology, Research and Development, and Dr. P.K. Upadhyay for giving me an opportunity uh, to be part of this webinar. Uh, when I joined and I had the previous lecture, I just joined in the end. So I was, wondering why didn't I join earlier? It was such an interesting talk. So I missed it, but I was busy in some other talk. So that's why I couldn't join. But it was really wonderful. And it's my pleasure to be here and to chair the session of where Dr. Rajendra Kumar is going to talk. I had the opportunity to see his passion from the ground itself. So he's a person who has been, you all know very well, he doesn't need any introduction. But I know as Director General, Uttar Pradesh Council of Agricultural Research, how he has bring out the transformation in that council, how he has contributed towards, improvising the agriculture practices there. So I have very close interaction with him, and I know his passion, and also about the science communication where he reaches to the masses, and that's why his contributions have been acknowledged. And this year I am very happy to share and talk about the President Award, which he received in March from none other than Dr. Ramnath Kovind, President of India. And I wish with his commitment to science, to promote science, he's going to bring about scientific temper in the society, which is needed at the moment. Uh, uh, I think we should just start with the lecture, and then how do you want to proceed, Anshuman?
Yes, ma'am. I would like to hand over the mic to Professor Rajendra Kumar sir, who is the Principal Scientist at Division of Genetics and Plant Breeding, IARI, Pusa, New Delhi, and also the former Director General, Uttar Pradesh Council of Agriculture Research, Upkar, Lucknow. Sir is the President of Indian Society of Genetics, Biotechnology Research and Development, Agra. Also, he is the President of various international and national societies. Recently, Sir has received an award from Honorable President of India regarding his work in science and technology. Sir would be giving a lecture on genetic gains in food legumes for human nutrition. I hand over the mic to Dr. Rajendra Kumar sir.
Thank you, Anshuman, Honorable Chairperson, Professor Ranjana Agrawal, guest, Professor Malika Dadlani, Dr. P.K. Upadhyaya. Anshuman, how I can share my slides?
Sir, yes, sir. You can go to share screen. Click on the share screen icon.
Yeah, I have done that.
Okay, no request has been received yet.
And not it?
No, sir, not yet.
I think now it has come.
No, sir, not yet.
No.
I think you have to select the screen and then press a button again.
Yeah.
Yes, got it. Yeah, here you are.
Yeah. It has come.
Okay, thank you.
Yeah.
Professor Ranjana Agrawal, Chairperson of the session, and Professor Malika Dadlani, former Joint Director Research, IRI, Dr. Upadhyaya, General Secretary, Indian Society for Genetics, Biotechnology Research and Development, Anshuman, Professor Bark, Professor Bahal, and many other learned colleagues participating as audience. Today I am going to deliver this talk, although I was preoccupied, but thanks goes to Dr. Upadhyaya. He compelled me that I should also give a talk. So my topic is related to my discipline only, genetic gains in food legumes for human nutrition. As far as food legumes are concerned, we all know that there are a series of food legumes like chickpea, pigeon pea, urd bean, mung bean, then lupins, many others. India is a major food legumes growing country in the world. They are grown about 23.5 million hectare with an annual production of 12 million tons. Even this quantity represents around 27.8% of the world production and puts India as one of the leading legume producing country globally. Pulses are still in the short supply, not only in the country, rather across the globe. Being rich in the protein, they not only form a vital part of the human diet for the vegetarians, rather even non-vegetarians also prefer that. Thus, pulses play a crucial role in balancing the dietary proteins. The cultivation of food legumes is not confined to the nutritional supplement. Rather, it has also added advantages for the succeeding crops. It is estimated that food legumes fix around 20 to 40 kg of valuable nitrogen from the atmosphere per hectare in the soil. Major food legumes, as I told earlier, that there are a huge number of major food legumes and minor food legumes. Major food legumes, Indian scenario, we are confined to pigeon pea, chickpea, the mung bean, urd bean, moth, lupins, etc. So lentil and many others. India grows the largest variety of food legumes in the world, sharing about 40% of the area and 33% of the total production. The major imported food legumes, legume producing states in the country are Madhya Pradesh, Maharashtra, Uttar Pradesh, Rajasthan, Andhra Pradesh, Karnataka, Gujarat, Chhattisgarh, and Bihar. These major growing states are cultivating at large scale gram, arha, mung bean, urd bean, lentil, peas, horse gram, and moth, etc. Food legumes are cultivated in India over a very large area and have been traditional crops. Even then, the area is fluctuating in not only area, rather production as well as productivity time to time due to various reasons. However, food legumes are important because they have several desirable nutritional attributes. Like they are very rich in the terms of the protein. They have good amount of dietary fiber. At the same time, they contain low fat, low glycemic index, and it's gluten free, high energy, source of micronutrients, Besides, it's a good source of anti-diabetic and anti-cancer properties, and it does not contain significant cholesterol. Thus, pulses, food legumes as pulses, commonly it is known as pulses in the common terms. So pulses are very important, and they function in various uh, way because they are nutritionally rich. They can be added in the value added products. Besides, they have several functional properties, and bioactive compounds can also be developed in addition to therapeutic values. We know that since ancient times, chickpea have been used in several ways, therapeutic values like beautifications items and face cleaning, accordingly bioactive compounds and several aspects. So I'm not going in detail because chickpea is one of the major crop amongst pulses, and India ranks second globally. As far as food legumes as a source of proteins are concerned, these are the very rich sources in terms of the protein, and these are often found in beans, lentils, chickpea, and peas, and 2-3 times higher in comparison to the cereals like wheat, rice, quinoa, oats, barley, and corn. Just taking one half of the lentil provides the almost equal amount of protein as compared with a cup of quinoa or 2 cups of rice or corn. If we compared with the animal and many other plant-based sources of protein, food legumes are more affordable and they are sustainable source of protein. Food legumes, besides protein, it's also good source of dietary fibers as well as carbohydrates. As they are excellent source of dietary fiber and complex carbohydrates, one cup of cooked food legumes, whatever maybe little variation, gives us more or less half the amount of fiber we need throughout the day. Food legumes also contain both soluble as well as insoluble fibers. Food legumes contain resistant starch, which is very important for control of fiber in body, and it also reduced release of the circulating cholesterol as well as maintains the blood sugar levels, and at the same time it also maintains the microbial gut in our system. Thus, let us have a look on the diverse nutritional value of various legume crops per 100 gram of the quantity. So we can see, if in case of protein, we see a variation. In case of soybean, it's on average around 18%. In case of peas, 8%. In case of lentil is 9%. In case of beans, 15%. In case of chickpea, 8.86%. In case of white bean, 9.73%, and in case of black bean, 8.86%. However, our studies have changed this data. Of course, this data is the USDA standard reference released on 28 food legumes. But we have observed that in case of chickpea, we can get protein up to 32%. So accordingly, there is a big variability, huge variability in terms of the other micro and macronutrients, whether we talk of the carbohydrate, dietary fiber, calcium, or whatever it may be. But one thing is established that food legumes are good source not only for the protein, rather other micro and macronutrients. Thus, we find there are major challenges as far as food legumes are concerned. In the light of the nutritional aspects like protein, carbohydrate, and other micro and macronutrients. Uh, the first one is the low genetic potential. Means the variation or variability which have been reported so far, it's very low in case of the pulses or food legume as a whole. Second bottleneck is the low realized yield. Pulses are supposed to be the poor yielder due to several factors. And uh, one more limitation is the climate change. Very climate change across the globe and uh, across the location also. Biotic and abiotic stresses. There are several biotic or abiotic stresses uh, across the different food legumes crops, ranging from salinity to drought, then heat stress, then in case of uh, biotic like Cardboard are fairly absorbed in case of pigeon pea, chickpea, and fusarium wilt in case of chickpea, rust in case of lentil. Like that, there are several biotic and abiotic stresses which influence. And finally, poor seed replacement, not only in case of the pulses, rather the whole crops. Professor Malika Dadlani will agree if I am correct. She might have gone through the one of our workshop conducted and analyzed in case of UP. We found a big gap as far as seed replacement is concerned. What to talk about the varietal replacement? I'm talking about the seed replacement. So that is also one of the major concern as far as pulses are concerned. Then post-harvest losses. Of course, these are supposed to be the less prone to the post-harvest losses. But even then, BGM or other post-harvest losses occur, and whenever the pulses are subjected to moisture condition, there is a deterioration in the quality and germination takes place. So that is also one of the post-harvest major reason. Poor transfer of technology. Ultimately, poor transfer of technology, one of the bottleneck, because these crops are very specific and they require specific agronomic practices as well as inputs in terms of the variety and region specific, climate specific, as well as requirement specific. So these technologies in terms of the food legumes are supposed to be efficient and they should have a wider access across the practitioners. Thus, challenges for the genetic gains for food legumes, especially in terms of nutrition. usually, so far we have been talking about the food requirement or food supply. Now, in the previous lectures, as Dr. Gupta has given an insight that right now country is sufficient as far as food supply is concerned. Rather, by the 2050, our requirement we are going to exceed from our resources what we have. But the problem is, so far we have been running after only the yield, our food supply, our food requirement. But now the change in the time requirement, food habits, and compelling malnutrition, particularly amongst children, women, like and pregnant women and elder people, have compelled us that we should divert our focus towards nutritional supply or nutritional requirement. But and we find that there is a negative correlation between the yield and quality traits as a whole, not only protein. Whatever nutritional traits, I can call it nutritional traits as quality traits. Why I'm calling quality traits? Because these nutritional traits are genetically governed by polygenes. And whenever they are governed by the polygenes, uh, then their control is very difficult. So we should not get here confused with the qualitative traits and quality traits. Here I am talking about the quality traits. Those are the nutritional traits basically. So there is a negative correlation between the yield and protein content. Mm, In addition to that, with the different diversified amino acids also, and the most important thing, so far we are not having proper screening technique for screening the large germ plant at length as far as nutritional traits are concerned, and that too integrated with the field activities or field screening. So these are the three major bottlenecks, and we have to come up these bottlenecks if we have to make certain genetic gains as far as food nutrition are concerned. Of course, negative correlation has been recorded in almost all food legumes species, but still there are certain exceptions showing party correlations and have been reported in several of the food legumes. Uh, those generated through the genetic recombinations or mutagenesis, like tillage population or like that, followed by selection. Once again, these are the building strategies we will be talking later. So future prospects for the genetic gain in case of the nutrition. Food legumes are deficient in sulfur-containing amino acids and tryptophan, but they are very rich in lysine. That's why we always call that let us eat in Indian our in the local language dal roti. So pulse and cereals they are good combination for integrating and enriching almost all essential amino acids for our requirements. Uh, Besides, we have to increase the biological value of food legumes. As far as biological value of food legumes are concerned, that's not big problem because it varies crop to crop, and it has very wide variation, ranging between 32 to 78%. Uh, of course, methionine content is low. That's why no biological value is there. But protein, in case of protein, we have very high biological value. In case of certain major crops of the food legumes, CP biological value ranges between 40 to 78%. Would mean 60 to 64%. Pigeon pea 46 to 74%. Lentil 32 to 58%, mung bean 39 to 66%, P48 to 49%. But out of these major food legumes, there are certain crops like chickpea, pigeon pea, lentil, and pea. These are having some anti-nutritional factors also in very high quantity. That's why some side consumptional problems also take place, like formation of the gas, or we call it those factors as anti-flatulence factors. We may discuss later. And that is the reason that the urd bean, moon bean are preferred crop, particularly those for those people who are ill or having poor digestive system. Bioavailable protein content in food legumes, in case of chickpea. So we have conducted one experiment in order to enhance the bioavailability of the chickpea. Of course, this is still under communication. We took 12 genotypes, and we took different parameters for the germination stages. So we found that 6 day germination stage was the best one for giving highest biological value for the protein in case of the taken variety. And the variety that was performing best is BG 1092, BG 1092. Uh, of course, it has outended the best performing variety Pusa 256 because that was used as control, and so far Pusa 256 has been considered as one of the very high protein container. Future prospects for genetic and breaking negative correlation between yield and protein. Choice for the breaking yield and protein linkage still exists because there are variability in the population developed by the mutation or natural mutants are through the mutagenesis like like dealing population or like that. So there is a chance because if we are not able to get enough variability in the germplasm of any food legume, we can go for mutagenesis also, development of the TD population. Of course, the rate of the variation is very low in case of mutagenesis because it's not directed at all. So, but still, whenever there is a mutation takes place, usually there is possibility that we can get one of the rare genotype which may have poor linkage between the yield as well as protein or other nutritional traits. Future process for breaking negative correlation between protein quality and quantity. I'm talking, there is a negative correlation that is particularly protein quality and quantity. So, negative. If the quality of the protein is higher, I mean to say biological value is higher, then usually quantity is the low. That needs to be broken. And of course, there are approaches which can be applied for breaking negative correlation between the quantity and quality of the protein and other traits under observation or preference. Better screening techniques for nutritional status of food legumes. Many procedures for chemical analysis of the protein as well as protein components like diverse amino acids, which we call 20 essential amino acids. Uh, there are techniques like IR reflectance and many other advanced techniques. Peptide analysis is one of the very important aspects. All food legumes are required to be exposed for the peptide analysis, and multi-mass spectrometry is one of the refinement which can take place. Amino acid analysis, protein structure, HPLC, detector IR, amino acid analysis system, Fourier transform infrared spectra, FTIR analysis. these are latest some equipments which can be applied, particularly FPIL over the SPLC, because so far SPLC have been utilized for these amino acid analysis and protein structure. Then comes the sugar and oligosaccharides. Of course, we can make these analyses through bioscan also, but we can analyze them through SPLC also. Uh, we used to take oligosaccharides, raffinose, utilizing SPLC, because oligosaccharides are very important. We may talk later. Mineral analysis. Besides, there are certain mineral analysis also, which are very important. So this can be analyzed through atomic absorption spectrometer utilization. Then ICP-MS, inductively coupled plasma mass spectrometry. Of course, very latest one of the version of this instrument has come. That is more refined one. Then finally, the improved field techniques which should be integrated for the use of genetic markers or molecular markers associated with the protein and other nutritional traits under observation or target. Genetic variability for nutritional traits in food legumes. Sufficient, as I indicated, sufficient variation exists for the main components in food legumes, ranging oil content from 1 to more than 40% and carbohydrate 2 to 60%. Thus, protein also varies 50 to 50% after drying weight. Manipulation can take place for these components, however, may not be, it's not easy task, because different combinations of these major components interact with each other in a complex biochemical mechanism, which mechanisms are very little known so far. We have to understand the mechanism involved for the interaction of these different nutritional traits also. I feel so far food legumes have been not been subjected to very intense studies as far as these traits were concerned. So there are not much reports as of course making basic profiling or proximate profiling for the nutritional composition is different, but knowing the pathway, then looking for their various components and utilizing those inferences for the betterment of the scientific output is a long way. Uh, Long back, we had conducted one of the protein analysis genotypes. Of course, we got 12 genotypes, so we identified one again. VG, PUSA 1092, 1092 gave the highest protein content, followed by ICC uh, 11378 genotype of the EQSET, which gave the highest protein content so far studied. But very recently, we are doing some more studies. We have observed 32%, but I am not reporting because still we need to validate. So besides, we have done also peptide analysis in case of protein. We subjected for the amino acids. Peptide analysis means different profiles of the amino acids. We took two genotypes of PUSA 1108, one of the Kabli chickpea, against one of the very old but still prominent Desi chickpea variety of Chandrasekhar Azad University of Agriculture and Technology, K850. So what we observed that these varieties were having very good amino acids, different amino acids, and we found these two varieties very close to the recommendations of FAO, WHO, as well as UNO. They talk about per day the per individual requirement. That is in case of threonine, cysteine, methionine, leucine, etc. So we find we can supplement these essential amino acids even with the limited quantity of the pulse. Otherwise, to, we have to look for the non-veg sources because in the cereals, these many of the amino acids are not available. Besides, these food legumes are also good source of antioxidants activities besides the nutraceutical profile and health relevant functionality of the certain genotypes of chickpea we studied, and we found that PUSA 1108 proved to be one of the very good genotype as far as antioxidants are concerned. We have already published it. Besides, we also made some studies for the enhancement of the bioavailability of different nutritional components of the chickpea in case of desi and chickpea. And once again, we found that different processes can play a role as far as enhancement of bioavailability of these nutritions are concerned. Uh, and we observed that as per Kabli chickpea is giving enhanced bioavailability in comparison to the desi chickpea. We have also published one report for the nutritional as well as anti-nutritional profile. Once again, we found that Kabli chickpea has performed better, and the nutritional aspects also that what that was found to be more biologically suitable for the human consumption, as well as nutritional supplement. We also studied for the nitrate reductase in case of the chickpea. What we found the, that reductate is a nitrate reductase is one of the enzymes which is very important for drought resistance, and we observed that GG11 uh, get the highest nitrate reductase with a value of 0.419. So these genotypes can be taken for the one of the source for drought resistance. Besides, we also studied superoxidase dismutase enzyme, which is supposed to be very good one of the enzyme which controls our cells injury, which is already proven in case of animal cells and of course in plant cells also. And we found that Icosat 3668, one of the genotype, proved to be very good as far as SOT enzyme availability is concerned. So that genotype can be uh, exploited for the SOT enzyme introgression in the agronomic varieties. Besides, we performed one experiment in case of oligosaccharides. I was talking about oligosaccharides whenever we talk of the anti-flatulence sectors or flatulence. There are three oligosaccharides, raffinose, barbascose statues. These cause very reason for the gas formation whenever we consume. So we made one of the study in case of chickpea, and we found that ICC 79 contained highest raffinose, and ICC 79 contains the lowest raffinose, and it can be very good for the just what, yeah, ICC 1683 can be used as one of the donor for the more of raffinose integration genes in economic variety. So we can mitigate the gas and other digestive problems. On the basis of our previous study, we tried to make a product also by incorporation of chickpea husk in case of the biscuit formation. And we found that it's giving very good output, and we are in the process of patenting. Let us see whether if it is patented. So now let us come to the genetic gain. Of course, as far as genetic studies in terms of the heritability, genetic advance, and other parameters in with specific reference to the nutritional traits are concerned, those have not been done at length. So far, very limited reports are there. Of course, so in case of soybean, we have report of 0.4 to 0.8% heritability, and in case of chickpea, 0.75 heritability, and in case of dry beans, 0.3 to 0.64% heritability as reported. So we feel that there is a need for the systematic study if we have to really address the FAO slogan that nutrition fulfillment. Because you know, has also in our sustainable development goal, given two goals, 1 and 3, that talks about the nutritional supplement in order to mitigate the death rate prevailing amongst children and adolescents, girls and women and elderly people. So whenever we talk of the genetic gain, it measures the genetic improvement or breeding progress and can be defined as the amount of increase in the performance over the parents through artificial selection, and is measured by difference between the population and their offspring. So this is one of the slide I'm sharing of Dr. Rajiv Wasle, one of the known, globally known scientists. He is the highest Google scholar as far as our country is concerned. So he has given some hypotheses that for harnessing this potential of the genetic gains, we have to focus on three aspects. First, we have to make the genotyping at the faster rate, and for doing the genotyping, we have to take the larger populations, and we should develop such a platform which reduces the cost of the experimentation or typing. At the same time, making selection of target years of season. And third, maintaining the favorable rare areas. And the last one, selecting years earlier on single plant basis. So finally, on the basis of all those things, what he he had to he advocates about a platform that advocates about the predicting the phenotypes in advance. That if, on the basis of parameters, considering the years per cycle, then genetic gain over the time and genetic variance, maintaining the genetic variance across the population, the germplasm, as well as years in terms of the selection intensity and selection accuracy, which is most important. Then including seed increase, multi-location trials to come up the heritability problems, and finally, these in toto can enhance our genetic gains. Another hypothesis given by published by Onbi Yu et al. In 2017. By and large, same hypothesis, but rather he had narrated different components like genetic variance, heritability, selection intensity, cycle time in various sub-components like germplasm, molecular markers, combination genetic parameters, case of genetic variation, population used for the estimation of the heritability, estimation of phenotypic variation, estimation of. So this slide talks about the more detailed activity about the previous slides in which Dr. Vasle talked about the four parameters. Accordingly, selection intensity, cycle time. So if we contribute all these things, these four parameters and subcomponents contribute to a larger output by different ways. Like different parameters, once again, we may discuss in other slides because here they are very small letters and I think visibility is poor, so audience may have problems. So, building strategies for genetic gains in food legumes for nutrition. I think there are four strategies. We should have a bigger program. I mean to say we should have bigger, larger population size. We should develop mechanized program, and we should develop an automated platform, and we should also integrate with the digital system. So we can dissect a larger population or gene pool for making assessment or our forecast future predictions for a particular trait. Next one is the adequate genetic variability. So we have to identify the trait-specific donors and elite but exotic materials. At the same time, we can go for generation of the genetic variability by uh, mutation breeding or other sources, ecotiling or yeah, other population systems. Then more accurate selection. For that, we have to very accurate. Our selection system is very robust. For that, our quality phenotyping, along with better experimental designs, number of replications, number of locations, and if we are able to integrate with the markers, then certainly we will be having enhanced high heritability. At the same time, faster breeding cycles because most of the food legumes either they have annual or perennial in their nature, like pigeon pea, and they take larger duration. So we have to shorten their breeding cycles. Either we can go for the speed breeding or other programs we can decide. Besides that, we can go for the genomic predictions also. Next one is the management that is empowered, which is very important. The management has to facilitate and should be accountable for product delivery. Only researchers cannot manage at length. In everywhere, every system is integrated with the management system, and the management system has to take care that all requirements are met out. Besides, well, well, trained staff who understand product development. We have to generate a battalion of the staff who should be very efficient and able to help the plant breeders by engineering tables, engineering operations, and other digital services and automation, etc. Uh, next one is the once again Dr. Vasle takes the thanks for this slide. Breeding strategies for genetic gains in food legumes for nutrition. Uh, of course, this slide talks at length very larger population. He talks about the gene bank, free breeding, then genomics and track discovery, forward breeding, say like molecular biology and genetic engineering, then finally seed system. Gene banks, we have to conserve the germ plant, we have to characterize, distribute the germ plants among the different stakeholders, and researches have to be carried forward. I also received long back code, mini core germplasm from the ITSET. We did many of the activities on those activities, and very recently last year I acquired core germplasm consisting of 1,000 genotypes. So we are dissecting those genotypes not only for the nutritional traits, rather other traits also, particularly nodulation traits and other traits. Then pre-breeding, like characterizing germplasm collection and making them suitable for breeding program, develop novel genetic populations, and at the same time we are also going for the mutation breeding also, tilling population. Then genomics and trade discovery, like genomic resources development and making comparative and functional genomic approaches for discovering the alleles responsible for a particular trait of interest. Or particularly, it can be compared with the Arabidopsis thaliana, which is already well dissected. The whole genome has already been dissected, and of course, medical setting also, these two genotypes have already been sequenced, and so they can be compared, and on the basis of their findings, we can identify the uh, uh, functional and operative genes for our target traits. Then we can move further forward building. Once we have done with the trade discovery and trade of interest, we can go further forward building. Besides molecular biology and genetic engineering, once again, there are number of approaches. We can go for different approaches. Can go for the applied development, transgenic, and other approaches also, cloning and other things. Then finally, the seed system, like once we have developed the system, we have to optimize and strengthen the seed delivery system. So it reaches the users at length. Hence, for seed production adoptions roadmaps in developing countries are required, and really it needs special attention, because whatever technologies we have already generated, those are not reaching to the farmers or end consumers. Then next, of course, many of the previous we have discussed. Even then, whatever building strategy we are adopting, those should be focused to climate resilience, nutrition focused, and market traits. How we can market, because ultimately farmers need economic strength. If we don't focus on the market requirement, that is also one of the failures, which has been always a problem with the farmers and producers. Then large-scale jump large sequencing and value addition, which we have already talked, that large-scale sequencing is required. Once sequencing is done, is all in case of all food groups, our task will be facilitated as far as any discovery and market association is concerned for different trait of interest for particular nutrients, whether it's protein or arginine or whatever it may be. Then breeding for pre-breeding, like our dealing population development or other compressed pre-breeding time and utilizing speed breeding or like that, whatever may be, but novel traits have to be identified through different approaches. Finally, we can go for the marker assisted selection and all those things. We can identify the QTLs, and diagnostic kits have to be identified for a trait of interest. Novel breeding approaches can be should be tested, validated, and deployed across the experiment, so faster delivery can be harvested. And biotechnology needs to be integrated with the crop breeding programs also, because biotechnology cannot deliver in isolation. So, including some biotechnology, other techniques and genomic approaches are required to be integrated and targeted for the nutritional traits. Of course, next one is the double haploid, still unexploited in case of the food legumes. There are certain problems, but this can also play a very important role as far as nutritional mapping and nutritional enhancements are concerned. So strengthening and modernization of the seed system, we have already talked. The new sequencing system, genotyping platforms, we have already discussed. Then resource mobilization, we talked in the previous slide also, enhancing capacity building and communication, which is lacking. We always talk about, but there are many things which we lack. There are no integration of the different branches of science. Although this is the era of the integration of the different science, not only science, rather we should be integrated along different streams of the science. Now the science definition has been changed. Even our disciplines are also defined as science. If we don't integrate our plant science with other um, art science, for example, economics or sociology, these are also sciences. All market data and societal data are generated through these art disciplines. So there is need to integrate our different um, uh, branch of the activities. Only then we can harness the potential of the nutritional requirement for the human as a whole. Once again, of course, just I am making duplication, but just in order to get a brain
Professor
better
Kumar, Professor
yeah.
Kumar, may I request you to conclude, please?
Okay, okay.
Thank
So I'm skipping this slide because we have already discussed earlier, and this also I'm skipping because there is no time. I don't think this is also required because we have already discussed, and please don't. So finally, we can say that we have to implant the food legumes of the nutrition by integrating the potential of the different disciplines of the science as a whole for the public health and nutrition. Biofortification is the requirement of the time. Special techniques like in vitro culture and producing these nutrients are required to be integrated. Mutagenesis is one of the potential area, of course, very limited scope, but it can be also integrated. So, the great potential of the several attempts in past to alter amino acids has been done, and this needs greater emphasis in future. Finally, all these studies can be integrated for a wider benefit of the society and particularly nutritional requirement. Once our nutritional requirement is met out, our immune system will be strengthened, and once immune is strong, certainly we will be able to encounter our pandemic situation, including our COVID-19. With all these, there are different categories, goes to more than 25 contributors across the country. And finally, I thank you all. If any questions, you are most welcome.
uh, Anshuman, are there some questions? Are there some
No,
questions
ma'am.
posted?
No, ma'am. No,
Uh,
ma'am. There are no questions. No
okay. Do you invite
questions.
questions?
Yes, we can invite questions.
Okay. So,
Any questions,
uh,
please?
Professor Kumar, uh, thank you for the wonderful talk. Uh, it is quite relevant, the genetic gains in food legumes for human nutrition, particularly in the present scenario, when in the COVID time, uh, you know, how things are going on, unprecedented, never ever seen before. And as you said, dal roti, you made a very bold statement in that, because when we see at the economy, the way it's going on, and particularly the marginal classes, how they have migrated, a mass migration has occurred, they have gone to the villages back, and even within the urban areas, how our food supply chain is going to be affected after COVID. So I think you have touched a very relevant topic, uh, uh, food legumes for human nutrition. because health is the most important component. And when I say health, it's not only the physical health, I'm also talking of the mental health too. And in India particularly, where most of the people, I will not say most, like majority of the people are vegetarian, so their protein content is basically dependent on either on dairy products or on legumes. And legumes are integral part of Indian human diet. I will say dal, banna, chawal, dal, rajma, chole, moong. ये हमारा food का एक natural part है. And also for the live food stock we have, and the ecological value associated with the fixation of nitrogen. So legumes definitely is the most important crop we are talking about. And you have very nicely talked about the benefits of legume proteins, like about the digestible proteins and low fat content, low cholesterol content, antioxidant activity, anti-cancer activity. All these things you summarized very nicely. And then you talked about the challenges for the genetic gains, future prospects. You talked very nicely, where you try to emphasize on like how we can break the negative correlation between yield and protein, and also how the negative correlation between protein quantity and protein quality. Some are the very important issues, because sometimes we have proteins in our diet, but we don't understand what is the quality of those proteins. Very nicely you talked about the amino acids, what are the amino acids which are present and what are absent, and how genetically modified legumes can give us holistic diet, more more nutritive diets. You also talked about screening, better screening methods, and definitely now we have very sophisticated instruments, so screening can be better and better. And the number of publications, it was so impressive, the work you have carried out with your genotype chickpeas. You have shown in your research studies about the the chickpea genotype, high protein content, amino acid profile, carbohydrates, antioxidant properties, nutraceutical properties. You also talked about enzymatic studies, nitrate reductase and superoxide dismutase. So I think all these research papers which you have published definitely show your commitment to this particular crop, chickpeas. And I have visited with you your fields, and how
Thank you.
passionately you showed me all your genetic mutant varieties. That was really really wonderful. And I really compliment you for the concluding remarks, where you said integration of different disciplines. It is really really very important. I think we have created verticals of agriculture sciences, medical sciences, physical sciences, biological sciences, and social sciences. And in silos we are working very nicely. But you raised a very prominent point, that integration of disciplines is very important, where we talk to social scientists also, economists also, and other scientists also, so that the technology which is being developed in one particular lab can be translated, can be transformed to the society in a big way. So I would like to congratulate you for such a wonderful talk. One or two questions I would like to ask.
Yeah.
One is a very general one, because I come from Haryana. And Haryana, Punjab, we generally see the rotation of two crops, rice and wheat. And we say like Haryana and Punjab is the uh, grain producer of the country. We feed the whole country. When we have so much biological, ecological significance attached to legumes, nutrition, which can give us a sustainable agriculture, less use of fertilizer, nitrates, etc. Even then, why our farmers do not get encouraged enough to go for crop alteration? This is my one question, because we all the time talk about crop rotation and all those things, but we are not able to motivate enough the farmers. I would like to take your views because you work on the field, you know better.
Yeah. Thank you, madam, for your compliments. As far as your question is concerned, it's a very good question and pertinent one. The reasons are basically two. One is that food legumes, for example, pigeon pea. In our scenario, it's a annual crop. It's not exactly, if not annual, it's a long duration crop. And the second one is that so far these crops have been cultivated with lesser inputs. The reason is that these food legumes are, by and large, all food legumes are indeterminate growth habit in their nature. So whenever the high inputs are given in terms of the fertilizer and irrigation, there is growth, huge growth, and there is no fruiting or part formation or yield. So there is a big gap. I mean to say, vegetative growth takes over the reproductive phase. That's why the high input applications are not applicable in case of the food legumes. Second reason. The third one is, of course, third, I'm giving on the priority wise. Third one is the baggage of the diseases and insects. There was a time when part borer had been one of the threat for the pigeon pea as well as chickpea both. I remember 2003, entire field of the IRI was infested with the part borer, and we harvested hardly a few limited soil seeds of our important genotypes. So, accordingly, drought. And fusarium wilt. So these are some. Fourth one, I cannot emphasize, but fourth one is the uh, lack of the proper support, and finally, there is a gap in the educational standard of the people, because we always run for the short goals. We don't take inference for the longer duration. You talked about the Punjab and Haryana. The reason is rice and wheat. They are intensive crops. And moreover, we have developed a number of short duration varieties. So rice and wheat, and with the high inputs in terms of the fertilizer, in terms of the irrigation. But you can see what is the situation of the soil fertility now. Now the same for Punjab and Haryana, they have started talking. No, we have to ban uh, rice, or we have to minimize the cultivation of the wheat. And even Punjab government has started giving incentive for the cultivation of the food legumes. So there is a need for the change. As far as farmers are concerned, still the economic conditions of farmers are not up to mark. Honestly speaking, that's not up to mark. They need their economic strength for want of the economic survival to meet out their daily requirements. They need some cash in hand. And in case of the pulses, so far it had been like that either insect pest devastated the crops, or sometime rains, heavy rains, that is also one of the high input. They also devastated the crop. And if anyhow there was the optimum rain, then insect devastated the crop. So there had been uncertainty as far as pulse or legume cultivations are concerned. That is the reason farmers also didn't like, due to some or other reasons. But now this is the requirement of the time, that we have to educate, because there are two added advantage. First one is the nutritional supplementation, the most important. Second one is it fixes the atmospheric nitrogen. None of other crop other than legumes fix atmospheric nitrogen at high rate in the soil. So I think these needs and farmers are required to be educated. Not audible. Yeah. Your mic, please.
Yeah, thank you for the comprehensive reply. You touched all the issues, and I'm sure with the policy interventions, as you mentioned, like enough support is not there, and with the good variety of genotype variety, which is drought resistant and pest resistant and all this, we will be seeing the encouragement in the farmers to grow those legumes. Thank you so very much. Thank you. Anshuman, you can take over.
Okay, thank you, madam.
Thank you very much, Professor Rajendra Kumar sir. Starting with the valedictory session, I would request the chief guest for today's valedictory session, Dr. Ranjana Agrawal ma'am, and guest of honor as well as special guest for today's valedictory session, Dr. Ulrich Berg sir, to have your kind words well to start with the valedictory session. May I proceed?
Sure, sure, sure.
Okay, thank you very much. I would now request Professor Rajendra Kumar sir to please do the compilation of the sessions of both the days as well as give the valedictory remark.
Honorable Chief Guest of the day, Professor Ranjana Agrawal, Guest of Honor, Professor Ulrich, Professor Malika Dadlani, I think she has left, and Dr. P.K. Upadhyaya, Anshuman, many dignitaries amongst audience, my colleagues, friends, students, and participants. We organized this 2 days webinar with objective to make a clear-cut inference how to benefit the pandemic situation uh, in the current scenario. We didn't like to confine only to COVID-19. Whenever we talk the pandemic always rolls. Sometimes some pandemic takes place, sometimes some. So anyway, so we uh, covered a number, very range of the deliberations. Yesterday, I want to say June 22, five deliberations were made. The first deliberations was made by the very chief guest, Dr. K.B. Prabhu. He narrated agricultural options and economic ventures with increased rural manpower availability. In the light of the government policies, how farmers and rural younger generation as well as migrants, particularly migrants from different parts of the country, they can be benefited from the scheme launched by the MSME, and how they can sustain their survival and their comfort. Besides, they can continue their other economic gain. And another lecture was very good lecture. Dr. Rawat delivered the prospects and strategies for the cultivation of medicinal and aromatic plants in the emerging situations. Not to, of course, very general, but not least important, because medicinal and aromatic plants have always taken place as a pivotal plants in the society. Uh, Somehow everyone needs the help of these medicinal plants, either as a allopathic medicines or as a naturopathy, or whatever you can say. But each and every medicines are the basically extracts of your plant system, except rare medicines. And aromatic plants also, they are very good in number, and they please our eyes, and at the same time he advocated that these can be suiting to our mindset also. Of course, the third lecture, of course, new technique like epigenetic variation. Now we talk about epigenetics. Epigenetic variation in response to early life exposure with reference to children health and diseases in human was narrated by Dr. Richard Safri, and no doubt epigenetics is the days of future, because this is one of the new science which is coming up. We have to understand the methylation process and all those things, and these new emerging technology can be exploited even in the plant system also. Next one, another lecture was given by Dr. Shweta Thakur regarding the atmospheric pollutions. She talked about phytoremediators with reference to the crop plants, and it was a good conclusion that plants should be encouraged, utilized, rather should come in practice to work as a factory for as phytoremediator. Another lecture, Professor I.S.G.B.R.D from AIMS. She talked at very length about the status of agriculture in the country and human health in the country in the light of the COVID-19 pandemic in the country. And of course, it was a very nice deliverance, and I agree with the remarks that there is a need to work, but I feel that there should be proper integration, interdisciplinary, transdisciplinary across the associations to various sciences, including social sciences. Today there were 6 lectures. One lecture in the morning that was chaired by Dr. H.S. Gupta, former DG Visa, and the lectures are given by Dr. Lakhbir Singh, Dr. R.K. Bahal, and Professor Abdul Sayur Rajak. They covered the area of the microbial electrotechnology, one of the new emerging technology, the very recent one in the nature. How to utilize those technologies for the utilization of the water energy for the benefit of the agriculture as a whole. Then another lecture was given by the food and health at length by Dr. Bahal. Once again, integrating the nutritional and health components together. Then Professor Abdul Razak talked about green technology, how green technology can play a role in maintaining the existing ecosystem, which ultimately leads to the mitigation of the pandemics in the society. And another lecture given by Dr. I.S.G.B.R.D Singh and Dr. Ulrich Berg was chaired by Professor Malvika Darlani, former Joint Director Research IARI. Professor Dr. I.S.G.B.R.D Singh talked about the human epidemiology and human health implications with reference to the Microbacterium in India. Besides, very interesting and enthusiastic lecture was given by the guest of honor, Professor Ulrich Berg, that how Agnihotra can help with the COVID-19. Honestly speaking, so far I was never impressed with the Agnihotra. But today, after going through his findings and deliberations, I seems to be convinced. So I have to looking more forward at how scientifically it can be enriched, and in the longer days it can come forward and accepted by the society at length for enriching the different prospects of the societal pandemics. And last lecture was given by me. I think my lecture has already been given by the chief guest of the session. So with all these things, I think this is the proceeding on the behalf of the society, and we feel thankful to the honorable chief guest and guest of honor and chairs of the respective sessions, and uh, dignified speakers during the different sessions, participants, and many of my friends, Professor Nanglal from Saudi University, Kanpur, and many others I cannot name. So, and dear students, with all these things, I hand over the mic to Ansuma, please.
Thank you very much, sir, for the conclusion of the session as well as for the valedictory remark. I would like to extend the vote of thanks. With this, we come to an end of our international webinar on challenges in agriculture and human health in emerging situations, held during 22nd and 23rd June 2020. I extend my heartiest vote of thanks and all our viewers who joined us live on this virtual platform. We reached a very good number of more than 2,000 viewers on both the days. That's a big milestone for all of us. I thank to all who got themselves registered in this international webinar and made us reach a very good number and a milestone for the webinar series with a good number of registrations exceeding more than 4,300. I would also like to thank Professor K.V. Prabhu sir, Chief Guest of our inaugural session, Guest of Honor, Honorable VC SKRU, Professor Rakshpal Singh sir, Special Guest, Professor M.P. Yadav sir. Our lead speakers for day one, our very own Chief Guest, Professor K.V. Prabhu sir, Dr. A.K.S. Rawat sir, Professor Richard Sefray, Dr. Shweta Thakur, and Professor I.S.G.B.R.D ma'am for their technical lectures on day one. I also extend my heartfelt thanks to the speakers of day two, Dr. Lakhvir Singh sir, Professor R.K. Behel sir, Dr. Abdul Shokar bin Abdurazak, Dr. Ajay Veer Singh sir, Dr. Ulrich Berg sir, Professor Rajendra Kumar sir. Chairperson of today's session, Dr. H.S. Gupta sir, Dr. Malika Dadlani ma'am, for sparing their valuable time from their busy schedules. I extend my sincere vote of thanks to our chief guest of this valedictory session, Dr. Ranjana Agrawal ma'am, and guest of honor, Dr. Ulrich Berg sir, for attending this valedictory session of this international webinar. I thank the webinar chairperson, Professor Rajendra Kumar Sir, former Director General UPCAR and Principal Scientist IARI New Delhi, our organizing secretary, Dr. P.K. Upadhyay, General Secretary, I.S.G.B.R.D, Chief Editor, Indian Research Journal of Genetics, Biotechnology Research and Development, Head, Department of Genetics and Plant Breeding, RBS College, Dr. B.R. Ambedkar University, and President, Dhadkan NGO, for their timely advice and all the guidance in the follow-up for this international webinar timely. Last but not the least, I bow down my head in the lost feet of the Almighty for showering His blessings all around during the preparations and commencement of this international webinar. As we have already reached the time limit of the session, I would like to make a few important announcements, which are: the Society is inviting research articles for our Indian Research Journal of Genetics, Biotechnology, and articles for our Hindi and English magazines Prakriti Prerak, Vishwa Krishi Uttan, Biotech Rainbow, and newsletter entitled Jal. We are also opening fellowship applications from 25th June 2020. Society will also announce some important awards in its upcoming international conference will for sure not will be a virtual media. People can also take memberships of the Indian Society of Genetics, Biotechnology Research and Development as soon as they wish. About the conference and the webinar, I would like to make a few announcements. All the e-certificates for the participants will be issued on attending all the sessions of this webinar, incepting from the day 1 till the end. Apart from this, the webinar feedback link will be shared on the WhatsApp groups created and will be shared by 23rd June 2020. The e-certificates to the participants will be provided after 20 days or within 20 days after the compilation of the webinar starting from 24th June. Thank you very much.
Thank you, madam. Thank you very much. So we can leave.
Yes,
Thank
we
you very
can
much.
leave. Thank you very much. Thank you very much.
Bye. Professor Ulrich. Nice to see
Thank
you.
you.
Yeah.
Thank
Okay,
you,
bye.
sir. Pranam.
Okay, Param. Everyone has left. Now we can talk or not.
Oh, no. Thank you.
So, Anshuman, you have to close.
Yes,
Yes, sir.
yes,
I'll close it. Thank you very much, sir.
Yeah.